Hello students, welcome to Vedantu NEET English. I am Ms. Gopika, your biology master teacher. Let me know in the comments and chats if I am audible, visible and the presentation right behind me is also visible. Let me know quickly, are we live yet? I think we'll be live any minute. Yes, we are live. Awesome. Yes, we are live. Hello students, how are you all? No, he will teach on this channel as well, okay? If he is live on JE also, he'll teach in this channel as well, okay? Yes, 1 second students. Some lights. Yes. Hello, hello. Shall we start the class? Okay, what are we going to do today? All of you, guess what are we going to do today? Today we are going to take some questions from some chapters in biology and we are going to solve it and we are going to understand what is actually happening, why this is right? So basically it's going to be like a PYQ session or a solving session where there will be some really easy questions and you will be solving that question. This is also to boost your confidence. At the same time, students, you will also understand where you stand, what you have to prepare, okay? So for zoology, I have taken questions from almost all the chapters that possible, but it is easy. Okay, easy questions, right? Yes, question practice, ma'am. Awesome. Did you all smash the like button? Let me know. And did you all share the link with your friends? Okay, I'm checking your attendance, all of you. No, all of you haven't marked your attendance. Until you mark the attendance, I will not proceed with the class. Okay, so quickly mark the attendance, all of you. It's very, very important that you mark your attendance as soon as you join the class. All of you, then I'll proceed. Okay, I should see. All of you mark your attendance. Then we will start the class immediately. And students, do understand that all the teachers will be taking the live in this video itself. That means in this link itself. You do not have to go anywhere, okay? After me, Baswa sir will come. After that, Shreya sir will come. After that, Diksha ma'am will come. So it will be continuous, okay? And in this span of this time, maybe you will solve easily 100 questions to 120 questions, okay? Which is an awesome thing because usually in the starting of the preparation, no one solves questions. They'll be like, it's okay, we will do it in the last. But this way, you'll also be practicing a lot of questions, okay? Chalo, I hope all of you mark the attendance. Let's start with the first question. The first question says that the functional unit of contraction of what? The functional unit of contraction of muscle is a portion of the myofibril between two successive Z lines, portion of the myofibril between two successive M line, center of H zone, center of I band. Let's see how many of you can answer. How many of you can answer? All of you try answering. How many of you remembers? Yes, try answering. Okay, some are telling option number 1, A for Ames. Okay. Okay, so what is it asking? The functional unit of contraction. So contraction, the functional unit is going to be your sarcomere. Now where is the sarcomere present? Between both the Z lines. Yes, between both the Z lines. So when the Z line moves forward is the reason why you are able to contract your muscles. Okay, done? Very good, amazing. Okay, starting was good, so let's keep it till the end. This energy should be there till the end. Keep on smashing the like button whenever you're joining, leaving, okay, all of this. Okay, it's very important. Chalo, so that is portion of myofibril between two successive Z line, that is called as your sarcomere. Okay, sarcomere. So when the Z lines move forward or moves inwards is when your muscle contracts. Shall we go to the next question? If this is the speed you're going to do, then I'll be done with 10 minutes only, okay? So this is the energy I want. All of you should answer. Those who do not know, learn it now, okay? Next one. Given below are two statements. The B peptide is not present in the mature insulin and it is removed during maturation into the insulin. Okay, this question comes from your biotechnology. I've taken all the high weightage chapters, okay? Statement 2, the main challenge for production of insulin using rDNA technique was getting insulin assembled in a mature form. So what are they telling? Here they are telling that out of A peptide, B peptide and C peptide chain, B peptide is removed. Which is right or wrong? You people tell me. The B peptide is not present in the mature insulin is what, okay, is what they are saying. B peptide. But I would say it's something else. It's something else. Okay. Removed during maturation into the insulin. Statement 2 says that the main challenge for production of insulin using rDNA technique was getting insulin assembled in the mature form. Okay, how many of you agree? How many of you disagree? Let's see. Okay, 1. A for amazing Gopika ma'am, A for Ammu ma'am. Okay, statement 1 and 2 are incorrect. Okay, some are telling 4. Okay, let's see what's the answer. Think students, see. In insulin, there is A chain, B chain and C chain. C chain will not be there in the mature state. But when they were forming insulin, they found it very difficult to make the whole peptide chain properly, to make the whole insulin chain properly. But they also found a trick for that. They were like, okay, when we are doing it in the matured form, why are we even thinking about the C peptide chain? Let's just remove it only, no? Only A and B should be there, right? Right. So, C peptide chain is not present in the mature insulin and it is removed during the maturation into the insulin. And this line comes from your NCERT itself. So, those students who read NCERT will know this line. So, what is the answer? The answer is going to be statement 1 is incorrect, statement 2 is correct. Student, one of the challenges, okay, statement 1 is incorrect, yeah, statement 2 is correct. One of the challenges that they faced while making insulin was to arrange this insulin. A chain is there, B chain is there, C peptide is there. They are not able to arrange this only. Then they were like, hey, wait, 1 second. During the mature stage, C peptide chain is not there. Then we will just remove it. Okay, we will just remove it. Got it? That was the thing. So, those who made mistake, you learnt it right now that if this assertion reasoning come, you are going to answer that statement, if the statement based question come, statement 1 is incorrect but statement 2 is correct. But instead here, if they give C peptide, then both the statements are correct. Okay, if they gave C peptide here, then both the statements are correct. Good to go? Yes or no? Yes, clear ma'am. Okay, chalo, next question. Assertion. During muscle contraction, I band and A band gets reduced. Reason, during contraction, thick filament slides over the myosin filament. I always tell you this, students, actin, thin filament. Actin, thin filament. Myosin, thick filament. Right, thick filament. So, statement 2 all of you know. Now, statement 1 you people tell me. During muscle contraction, I band and A band will get reduced. They told I will not do any work. If Z line wants to get reduced, you reduce. Okay, if M line wants to do something, you do. I will not move at all. Who are the people who told that? I band and A band. Yes or no? Then, during contraction, thick filament slides over myosin. Who is thick filament? Myosin itself. So, myosin will slide over myosin. No, actin will slide over myosin. Sliding filament theory, right? So, what is going to be there? Yes, exactly. A band is constant. D for doctor. Answer is going to be D for doctor. That is both A and R are false. Any doubts in this? Any doubts in this? Yes, they don't get reduced. Exactly. They don't get reduced. They are like, whatever you want, you do. Like I am telling, no, students like the session, come in students, whatever you want, you tell. I am constant. I will not listen nor I will do. Some students are there in the class like that. So, same way this I band and A band is. Are we good to go? Did you all mark the attendance? Trisha, whatever we are doing in this channel is going to be for NEET 2025 itself. NEET 2024 got over. So, we are preparing students for the next year, right? Awesome. Next question. What is fascicles? Connective tissue layer, epithelial tissue layer, muscle bundles, group of myofibrils. What is fascicles? If this is the muscle, some groups are there, bundles are there, muscle bundles are there. Inside that muscle fibers are there. Yes or no? Muscle fibers are there. Who is this fascicles? All of you tell me. Who is fascicles? Any idea? connective tissue layer, epithelial tissue layer, muscle bundles, group of myofibrils. Even if you don't know this, by looking at this you can tell, because I told the answer. I told the answer. Yes, C for capto. Very good, very good, awesome. The answer is going to be, they are muscle bundles, right? So this is your muscle. Inside that, muscle bundles will be there, right? These muscle bundles are called as fascicles. These are muscle fibers, right? And these muscle bundles will be kept together, right? They will be kept together with the help of fascia, okay? Like this one, fascia will be there, which will keep all these muscle bundles together. Agreed, all of you? Very good. I think this was an easy question. Next one. Oh, looking at the question, you'll be like, oh my god, so difficult. Not at all. They're asking you to look at this diagram and tell which is the option. Okay, which is the first one here? A is given. A, B, C, D, E is there, students? E. Okay, this is what they have asked. So if you look here, you can see that myosin head is there, actin is there. Cross bridge formation has happened. I told you it's like two countries, no? Cross bridge will happen. When actin and myosin come together and bind, cross bridge will happen. When they move out, power stroke will happen. That means the ADP and PI will forcefully move out, and that will cause the power stroke. Correct. Now let's see which are the options that is right. A, let's say cross bridge. What will be B? Any answers? Check properly. Take your time, no problem. We have C, we have D, we have E. Okay, very important question. Very, very important question, but at the same time, very easy concept also. Okay, Mukunda is telling 3, option number 3. Okay. Yeah, fine. Other people? Other people, all of you should tell. Only one student tell, how will it happen? 3, option number 3. Let's lock option number 3. Shall I lock? Okay, 3, fine. Let's see. Very good, because very simple, students. Here cross bridge happened. Whenever actin and myosin, see, if you want to do muscle contraction, two people are there, actin and myosin. When they come together, cross bridge formation will happen. When they move, that means when this myosin head, ATP is there, ADP and PI is there, when they try to move, sliding will happen. After that, again cross bridge will break. Okay, this is the same thing. So look here, A is cross bridge, B is cross bridge formation, C is sliding and rotation, D is breaking of the cross bridge, E is ATP. Whenever new ATP comes, the cross bridge will automatically break. Okay, right? Whenever the new person comes, that friendship between two people will always break. Not like that. This is just, just an assumption. Okay, next question. Which one of the following is correct match? Who is matching with each other? Okay, making RNA from RNA, transcription. Making RNA to DNA, reverse transcription. Making DNA to DNA, translation. Making RNA to protein, replication. Okay, if you all remember the central dogma, we know this. DNA to mRNA, mRNA to protein, okay. DNA to mRNA, one name is there, mRNA to protein, one name is there, DNA can form its own replica, one name is there, mRNA to form DNA also, one name is there. I am not giving clues. Okay, I am not giving clue. This is an easy question. You have studied this in your smaller classes as well. Lock option number 2. I'm locking option number 2, that is making RNA to DNA, which is called as reverse transcription, and the enzyme used is reverse transcriptase, right? Normally DNA to mRNA is formed, that is transcription. Whenever it goes from mRNA to DNA, it will be called reverse transcription. Easy? Easy? Very good. Good job. How many of you got everything right so far? Tell me your names in the chat. How many of you got everything right so far? Quickly, yes, central dogma. The ruling of molecular biology is because of the central dogma. How many of you raise your hands or put fire emoji if you got all correct? So far, all correct. Or at least maybe 4 correct? Fire emojis if you have got it. Let's see how many fire emojis are going to come. Even if you made only one mistake, it's fine. Just put Okay, you'll raise your hands or fire emoji, any one of it. Or put that pink hand. In the YouTube is there, no? Pink hand. One wrong. Okay, no problem. Put some emoji, people. What is this? Ha, very good. Ha, now I can see. Good job. Okay. See, at this stage, if you are able to solve, that is a good, it's showing good, okay, that you can do. You will be capable to do by December. You will be good at the entire topics, okay, if you are following us. Okay, yes, next question. A short length of DNA molecule has 80 thymine, 80 guanine bases, the total number of nucleotides. So, we all know that nitrogenous bases are there, purines and pyrimidines is there, and they are going to bind how? At Gopika ma'am's class, right? At Gopika ma'am's class. At Gopika ma'am's class. This is how they'll bind. How many ever adenines is there? That many ever thymines will be there. How many ever guanines is there? That many ever cytosines will be there. So let's see what's the answer here, students. Here they're telling 80 thymine is there. Okay, A we don't know. Okay, A is question mark. A is question mark. They're telling thymine 80 is there. Then they're telling guanine. Okay, guanine they're telling. Okay, wait. Here they're telling 80 is there. Cytosine is question mark. Obviously, students, in a class 10 people are there. I'll ask you to become groups. You will become a group of 2. Yeah, it will become group of 2. If more number is there, then one person will be left out. Same way, how many ever adenine is there, that many thymine should be there. So, if I do it, it's very simple. 80 is equal to 80. 80 is equal to 80. So, what will be the total number of nucleotides? Total number of nucleotides. Let's see your maths, if you are weak in maths or not. Those who did not mark attendance. Students, why you people are not marking answer? Attendance. I can't even tell that word. Repeating. Okay, nice. Madhumita is coming with new words. Dynamic. Okay, very nice. What's the answer? 320. 320. Yes or no? 80. 8 4s are 80 into 4. 320. 80 into 4 is 320. Correct? Correct or wrong? Done. Let's see if the answer is correct. Perfect. Okay, next question. Oh, students, it's so bright. Yeah, you people can't see this. Wait 1 second. What is happening? Yeah. Okay, do this, then we'll come to that question. I'll adjust the brightness. Okay. Which cells of the body is known as HIV factory? Okay. Human virus, right? Immunodeficiency virus. This virus causes AIDS. All of us know this. But it will get into one cell. It will get into one cell and it will multiply, but it will never kill that cell. Which is that cell? All of you thinking, tell me. HIV will get into one cell. It will keep making copies, but it will not kill the cell. But at the same time, if this cell goes to a T cell, it will kill that T cell. Which is that cell? Lymphocytes, eosinophils, basophils, macrophages. When I taught you body fluids and circulation last week, I spoke about all these 3 people. Did I speak about macrophages? I did not speak about macrophages. So, who is going to be the answer? Not because I didn't speak. It's because macrophages are the factory. Like, it's like a factory of HIV. It will not kill macrophages. It will not kill macrophages. Okay, it will keep producing factory. That means it will keep making its viral proteins. Continuously, it will keep making the copies. Okay, so the answer is going to be macrophages. Okay, I locked the answer. The answer is correct. Which among the following is used to treat emphysema? This also I told you in breathing and exchange chapter. I told you that emphysema, there is one human, I will not tell the answer. Someone is there, or some person, it's not a person, something is there that would actually help us treat emphysema. But a person continuously smoking, whatever was in the body would have, vanished. But we will use transgenic animals to form this. What is that answer? What is that answer? A, some people are telling, okay. Check properly, okay. One word is there. One word is there. We'll see how many of them makes mistake in this. Yes, one word is there. Tell me what is that word. Okay, I'll move. Yeah, no removing the messages, okay? Yes, biotechnology is there. Yes, Madhumita, Krishna, okay. Okay, some of you. Okay, fine. Rajashekar, okay, fine. Look here. Human hormone is there, human protein is there. I will go with protein. Okay, human protein that is called as alpha-1 antitrypsin. This is the one that can treat emphysema, and I will use a transgenic animal. I will use a transgenic animal to cure this. Animal to cure this. Got it, all of you? The transgenic animal name is called Tracy. So it's a human protein, it's not a human hormone. When I teach you chemical coordination, have I ever told you about alpha-1 antitrypsin? No, it's a protein. Okay, it's a protein that can treat emphysema, but a person, because of continuous cigarette smoking, be it passive or active, that person will not be, the body will not be able to cure it. So it will become a chronic disorder. So we will use another animal, which is a sheep. Okay, Tracy, the sheep name is. We will use that animal to form new alpha-1 antitrypsin. Done? Concept also I am telling and going, so this way you will be clear. Okay, one very common question, but majority students make mistake. Radial symmetry is not found in adults of dash phylum. Okay, Hemichordata, Coelenterata, Echinodermata, Ctenophora. So radial symmetry is not found. Not found. See, not word is capital letter. Yes, Tracy is the sheep. Very good. Okay, answer this, students. Answer this. Radial symmetry is not found in adults of phylum Hemichordata, Coelenterata, Echinodermata, Ctenophora. Charm will go now. Charm will go now when you see the answer. Think properly, people. Echinodermata, larval stage will have one, adult stage will have one. What does adult stage have? Does it have radial symmetry or does it have bilateral symmetry? Okay, fine. How many of you this is a question that forever can be confusing for a student. Forever. How many ever times I do it? I think I've done it so many times on the channel, but still it's confusing. Okay? Yes. Very good. The answer is going to be Hemichordata, not Echinodermata, because Echinodermata adult stage will have radial symmetry, larval stage will have bilateral symmetry. This is just to confuse you. This question is existing just to confuse you, okay? And this might be that one question that has come from animal kingdom, right? So you have to be carefully listen to me. Echinodermata adult stage will have radial symmetry and the young ones have bilateral symmetry. Okay, Echinodermata word is given just to confuse. Okay, otherwise why would they give you Echinodermata out of all of them? So in Echinodermata, two things are there. Adult will have radial symmetry and the larval bilateral symmetry. Got it? Make a note. Now only take a paper and write and keep. Okay? So the answer is going to be, okay, fine. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? This is also a very repeated question, but a very simple basic question. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? Okay, this question is asked. This point is there, Rani. Echinodermata adult and larval stage is there. But this point as such will not be mentioned. Definitely one line of NCERT is there telling that Echinodermata in adult stage it's radial symmetry, in larval stage it's bilateral symmetry. With that they have made a question. Okay, to confuse. To confuse. Okay, don't feel depressed, yeah. What is this? As if you lost the World Cup. Just one question, no? We are learning. We are not trying to be champions today. It's a slow process. Right? After 6 months, if I do the same thing and you make mistake, that time I'll scold you. Now I am teaching you. So it's fine. Chalo, tell me this. So students, I have always told you, remember like this, R, M and T. Okay, this one RNA, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Okay, these are RNA polymerase. RNA polymerase 1, 2 and 3, and rRNA, mRNA, tRNA. This is how you'll remember. This is the trick to remember RMT, like your MTR, masala and all. No? Remember RMT also. So what's going to be the answer? What's going to be the answer? What's going to be the answer? All of you tell me. Yes. A is the answer. Very good, very good. A is the answer because here you can see, no, tRNA. tRNA. Their question was RNA polymerase 3. So according to our R, M and T, this is 3. Right? 3. tRNA. Okay, that's how you remember. Don't forget the question. Next one. Which was the last human chromosome to be completely sequenced? Completely sequenced. Which was the last human chromosome to be completely sequenced, but the number is kind of first. Okay, I've given you the trick. I've given you the clue. Now with the clue, just answer. Yes, try answering. All of you, fast. I've given the trick so that it's easy actually. Chromosome 1. Okay, not bad, yeah, not bad. People got the hint. People got the hint. Yes, exactly. So this is an easy question. Why? See, which one was the last chromosome? But if you see the number, it is chromosome 1. Usually students will not think like that. Students will be like, hey, how can first chromosome be last sequence? But actually, according to our HGB project, the entire sequencing, chromosome 1 was the one that was last sequenced. Okay, so you remember like this always. Last one which was sequenced is the chromosome number 1. Okay. Yes. Next question. DNA replication is conservative and discontinuous, semi-conservative and semi-discontinuous, semi-conservative and discontinuous, conservative. Okay, that is, what is that? This is the parent. These are the parent DNA, and this will be the daughter DNA or the new one that will be formed. How do our DNA form, students? Replication. One parent DNA, one daughter DNA. So it is semi-conservative. But other than that, one more word is there. Try to find that word, all of you. Is it conservative and discontinuous? No, ma'am. It is going to be semi-conservative because one parent DNA will be there, one daughter DNA will be there. Their proof was Messelson and Stahl experiment where they took N15 and N14. One heavy DNA and one light DNA. Okay, one heavy, one light. Hybrid will be formed. Now the next question is, what will be, will it be semi-conservative, semi-discontinuous, semi-conservative, discontinuous? Okay, some of them got it. Usha, Hasini, Shweta. Students, those who are new to the channel without subscribing, do not leave because you get so much content in this channel, be it about your counseling, be it about your selection, be it about your college, be it about the NTA scam. Every single thing, and amazing classes also. Every single day live is happening. Every single day one-shot classes are happening. By parts also we are doing. So I don't think you should ever forget subscribing to the channel. Okay, not forget to subscribe to the channel. It will actually help your preparation. Yes, very good. The answer is going to be B, that is semi-conservative and semi-discontinuous. Okay, both are going to be semi-discontinuous, semi-conservative. Okay, got it? Next one. Identify the correct statement with respect to lac-operon. First one, regulation of lac-operon by repressor is referred to as positive control. Second one, operator gene has a site for binding of RNA polymerase. LAC-Z gene codes for beta-galactosidase. Okay, you have to find the correct one. Okay, I was like, how come everything is wrong? Okay, correct one you have to find. Got it? Yes. Look here, regulator gene binds with repressor protein molecule, prevents transcription of structural gene. Okay, now students find the correct one. Okay, find the correct one. Regulation of lac operon by repressor is referred to as positive control. Operator gene has a site for binding of RNA polymerase. LAC-Z gene codes for beta-galactosidase. Regulator gene binds with repressor protein molecule, prevents the transcription of structural genes. I think, I don't know if I spilled the answer. I don't think I spilled the answer, no? Yes. What's the answer? So LAC, that is going to be LAC-Z, LAC-Y, LAC-A. This is going to release beta-galactosidase. This is going to give permease. This is going to give transacetylase. Right? Transacetylase. So which is the correct answer? Obviously option number C, that is LAC-Z gene codes for beta-galactosidase. Awesome. Okay, very good, very good. Now next one. Angiotensin 2 increases the glomerular blood pressure, thereby GFR. Angiotensin 2 activates the JG cells to release renin. So students, angiotensin 2 increases the glomerular blood pressure, thereby GFR. Angiotensin 2 activates JG cells to release renin. What is the answer? If both assertion and reason are true, reason is the correct explanation, not the correct explanation. One is true, one is false, one is false, one is true. You people know this answer because that fellow cannot do anything individually, so he'll go and activate someone. That person will activate another person, so it continues. So let's see which is the answer. Angiotensin 2 activates the JG cell to release renin is what they're telling. I thought macula densa was going to activate or give that signal to JG cells and then JG cells will release renin. Students, if there is a gutbud in the GFR, glomerular filtrate rate, who is going to sense it? One quality checking people will be there. No, that quality checking people will be who? Macula densa in the DCT. That macula densa will tell JG cells release renin. Some problem is there in the GFR. This fellow did not drink water. This girl did not eat any salt from morning. I have to do something. So who is going to do that? It is going to be macula densa, not angiotensin 2. Angiotensin 2 is the last person who can fix all the problems. The fixer is angiotensin 2. So what's the answer? Okay, D, B, C, D, everything. Answer is C. Okay, if assertion is true but reason is false, this is absolutely false. Right, because macula densa, okay, in the DCT will activate JG cells and then JG cells will release renin. Correct? So this is wrong. All of you got it? Those who made mistake also got it? Why it is not right? Okay, this is the angiotensin 2 is the last stage who will actually fix something. Okay, got it? Yes, fine. A fall in the glomerular filtration rate activates. See, question came off. A fall in the glomerular filtrate rate activates juxtaglomerular cells, that is your JG cells. Okay, don't see the big name and faint. Simply known as JG cells. Juxtaglomerular cells to release renin, adrenal cortex to release aldosterone, adrenal medulla to release adrenaline, posterior pituitary to release vasopressin. My God, all are ready to release something and fix the GFR, students. But who is the actual person who is going to do? So many fixers. Okay, so many fixers here. But let's see who is going to actually fix it. Is it Ammu ma'am or is it Diksha ma'am? Is it Capto? Is it Baswa sir? Let's see who is going to fix this. All those who joined right now, quickly mark your attendance. Okay, mark your attendance. Okay, Gopika ma'am is going to win, na? Yes, Gopika ma'am did win. Okay, Ammu ma'am is winning because first JG cells have to release renin. Renin will go and activate angiotensinogen. Okay, renin will activate, should I write it with another color? Okay, angiotensinogen. This angiotensinogen will activate angiotensin 1. Angiotensin 1 will activate angiotensin 2 with the help of ACE that is released by the liver. That is angiotensin converting enzyme. Done? Done? Very good, okay? Yes. During micturition, that means when urine is expelled out, urine is prevented from flowing back into the uterus. Urethral sphincters contract during micturition. What is happening? Okay? Yes, Hasini, always. Okay? Chalo. During micturition, urine is prevented from flowing back into the uterus. This is what the first statement is. Reason is, urethral sphincters contract during micturition. I have told you this very clearly to you people. Urinary bladder has to contract for the urine to be expelled out. Right? Yes? Yes, my favorite chapter. Okay, so urinary bladder has to contract for urine to be expelled out. But the sphincter muscles will have to contract or relax. Contract or relax? Okay, I think some of you got it. Urinary bladder contracts, sphincter muscles have to relax, and then the urine will be expelled out. If they contract, it will be closed. They have to relax. They'll be contracted when we don't have to expel out. That means when the urinary bladder is still filling. Okay, it can go up to 800. So till 500, 600, you can control. Okay, yes, very good. Relax, ma'am. I should relax, sir. Sphincter muscle should relax. Okay? Yes. Very good, very good. All of you got the answer. Let's see. Let's see. Answer is perfectly right. If assertion is true, reason is false. Exactly, because here urethral sphincters have to relax for the urine to be expelled out. If they contract, then closing is going to happen. Okay, so the answer is going to be option number C. Very good. Next question. With reference to pituitary, which of the following statement is correct? Pituitary gland. Okay, pituitary gland has three portions: anterior lobe, middle lobe, and the posterior lobe. They can also be called as adenohypophysis, neurohypophysis, right? This all of you remember. Now, with reference to pituitary, students, there might be a lot of students who forgot this. Like, a lot of students will be like, ma'am, I do not know this. What do I do? Do not worry. Okay, this class will help you understand, okay, I can solve questions like this. If I made mistake, I will correct myself. But do not miss this session. Okay, because 100 questions doing one day in a span of maybe 3 hours is good, because the teacher will also explain you the concept. You'll understand the concept. You'll also know where you are weak, where you have to study. So that's all. Then later watch the classes and then you will be sorted. Okay, yeah. Which of the following statement is correct? Neurohypophysis synthesizes and releases vasopressin and oxytocin. Okay, statement is correct. Okay, we want correct statement. Adenohypophysis stores TCH secreted by the neurohypophysis. Pars nervosa stores and release vasopressin and oxytocin. Pars distalis secretes, everyone are behind vasopressin and oxytocin, students. Okay, so understand, there is anterior lobe, okay, anterior lobe, posterior lobe. Posterior lobe will release only 2 of them. Only 2 hormones will come from the posterior lobe. That is your vasopressin and oxytocin. Pars nervosa is there, pars distalis also is there. That is again other names. Anterior lobe, posterior lobe, middle lobe, different names have been given. Let's see how many of you guess the answer right. Okay, okay, very good, very good. That is option number C. Pars nervosa, again your posterior lobe, will store and release vasopressin and oxytocin. Their neurohypophysis synthesizes and releases vasopressin and oxytocin. Oxytocin is not right, because hypothalamus have neurosecretory cells that come all the way down to the posterior lobe and secrete their secretions there. So who is going to release? Storing and releasing will be done by the posterior lobe. Is this clear, all of you? Is this clear, all of you? Many of you would have thought, okay, option A is correct because posterior lobe other name. But understand, Your hypothalamus is like the principal. Other people are like the teachers, okay? So principal will have neurosecretory cells that will bring the secretions and release it there. Then their job is to store and release. Got it? Fine. Yes. Very nice. Next one. Dash is the protective envelope around the ovule. Dash is the protective envelope around the ovule. How come botany questions came, students? That's what you're thinking, no? Oh, this is, I have put from sexual reproduction also, okay? Don't take tension. I taught you sexual reproduction, so I have put some questions from there, okay? It's zoology and botany, biology it is. Dash is a protective envelope around ovule. Very easy question. Very easy question. Hilum is there. Hilum is the place where the funicle attaches with the ovule, okay? Funicle and ovule get attached, that is the hilum. Then you have placenta. Okay, then you have placenta is the one where the ovule attaches to the ovary. Then you have integuments which protects. Then you have style. I kind of told the answer, but yeah, it's okay. Tell me. Very good. So students, if you all remember, this was our ovule, right? This is the ovule. Wait, I'll draw the funicle this side. Right, this is the ovule. This is the ovule, right? So here funicle is there. This inside this embryo sac will be there. Embryo sac will be there. Who is this people? Integuments that actually protect, right? That actually protect. So option number C, that is integuments, is the answer. Good job. Okay, good job. Next one. Pollen grains lose their viability within 30 minutes of their release. I taught you this yesterday. Pollen grain loses their viability within 30 minutes of their release in rose, potato, wheat, pea. I told you, viability is how long this pollen grain will be active for fertilization to happen. It has to form its pollen tube, go all the way to the stigma, from the stigma to the ovary, right? How long they will be viable? So we learned few of them for 1 month, few of them for 30 minutes. Let's see. Thank you, Krishna. Yes, tell me the answer. Pollen grain lose their viability within 30 minutes of their release in rose, potato, wheat and pea plant. Yes, very good, very good. All of you remember? Amazing. That is wheat, right? Wheat. 30 minutes is rice and wheat. 1 month, students. All of you answer me. 1 month. Rice and wheat. 1 month. Almost all the options are here, okay? 1 month, who is there? 1 month viability, who can have? This is 30 minutes, I agree. 1 month? You can tell me the family names also, no problem. Who are they? Rosaceae, Leguminaceae, Solanaceae. Solanaceae. How will you remember? Rosole. Rosole में क्या है? Something like that in Hindi it was there, students. I forgot. Rosaceae, Solanaceae, Leguminaceae. These three families will have, the pollen grains will have a viability for 1 month. Okay? Chalo, next question. In the sewage treatment, bacterial flocks are allowed to sediment in the settling tank. This sediment is called as, see, you are revising almost all the chapters. All the chapters. Okay? In this question, you will be revising everything. You will be remembering, you might forget some things. That's why practice sessions are very nice. In sewage treatment, bacterial flocks are allowed to sediment in a settling tank. This sediment is called as inactivated sludge. Who is settling? Bacterial flocks. Activated sludge, primary sludge, secondary sludge. Give it a try. Even if it's wrong, it's okay. Give it a try. Yes, wheat is correct. After wheat, please proceed. Wheat. Yes. Yes, correct. Okay. Give it a try. In sewage treatment, bacterial flocks will be settling. Okay, and that sediment will be called as what? Tell your answers. Wrong, right, doesn't matter. We are learning, so it's okay. Okay, activated sludge, primary sludge. Many of them have confusion with it, within activated sludge and primary sludge. Okay, which do I lock the answer? Shall I lock option number B, activated sludge? Students, this is the activated sludge because bacterial flocks, right? Your aerobic bacteria are there to consume any organic material that is there. They will use up the organic, or they will eat up that organic material, so the water will go for the next stage. So activated sludge is going to be the answer whenever bacterial flocks are coming, right? So primary treatment is there. Primary treatment will remove all this plastic cover, cigarette, that, this and all. When secondary treatment come, we'll give this aerobic, that sludge we give, right? So activated sludge. Next question. Yes, Monascus purpuris is used commercially in the production of ethanol, streptococcus for removing clots from the blood vessels, citric acid, blood cholesterol lowering statins. Okay, Monascus purpuris. Students, these are few microbes. Only one question will come, but these are the few terms that will always come from, you know, microbes chapter. That is, Monascus purpuris is used commercially, okay, commercially in the production of ethanol, streptococcus, citric acid, blood cholesterol lowering agent. Yes? Tell me the answer. D, ma'am. Blood cholesterol lowering statins. All of you have locked. Very nice, very nice. Okay, okay, let's see the answer. Perfectly right, right? Whenever a person goes with a cardiac arrest, or a person goes with any heart problem, and the doctor understands that it is because of an accumulation of a plaque, or cholesterol has been deposited. Now students, normally what happens, this is your vessels, right? Your arteries are there. Now in the arteries, because you consume a lot of red meat, or you consume a lot of, you know, junk food, but you don't exercise enough, then accumulation of fat will happen here. Okay, accumulation of fat will happen, bad cholesterol basically. So a person, that is the doctor, will immediately give, yes, immediately they will give a Monascus purpuris. That means any blood cholesterol lowering agent, okay, lowering agent, which will actually control the whole situation. Okay, yes. Next question. Oh, this question has been repeated, students. 2024 also similar question came, but the options were that side, this side different. Okay, very favorite question of NTA. I mean, yeah. Pectinase and protease, streptokinase, cyclosporin A, and statins. Now you already know statins: blood cholesterol lowering agent, immunosuppressive agent, clot buster, clarifying agent. I have asked you clot buster so many times, and even immunosuppressive agent. Okay, answer. See, if you know at least two options, we'll be able to answer. Say, I know one option, okay? That is this one. Say, I know this one option. Student did not study, they only know one option. Let's see where C2 is there. Okay, students, you 50-50, okay? This is Kaun Banega Crorepati. So, C2 is seen in this 2. So, I will cancel this also. I'll cancel this also. 50-50 done. Now, you have to have confusion between B and D. Confusion should be between B and D. This is how you do match the following. Okay, so you know this also, okay? Which one else you know? Clot buster, you know? Clarifying agent, you know? Okay, clarifying agent, you know? A4. Oh, I got the answer. I got the answer. B is the answer. Yes, yes, yes. Okay, so you can do this 50-50, phone a friend. You cannot phone a friend, then you will be debarred. But you can do this. Two options, you know. See, with two options, I did the whole thing. I did not even know all of the options also. I'll be able to do this. See, streptokinase, clotting factor, stannin, blood cholesterol lowering agent. Okay? Yes, done. Was that easy? Yes, awesome. Next one. Oh, this question came, students. PYQ, repeated multiple years. PYQ. What is this? Aspergillus niger, Clostridium butylatum, Saccharomyces cerevisiae, your yeast, and Trichoderma, ethanol, citric acid, butyric acid, cyclosporin A. Cyclosporin A, now you know. Yes. Answer this. Let me see if you'll use the same brain. Students, will try 50-50 if you want. Okay, try 50-50 if you want. Yeast. Think of yeast. Ethanol. Okay, that is 3A. 3A. Ah, okay, students, 50-50. Okay, option number B and C. B and C. Okay, now in 50-50, which one do we know? Clostridium butylatum, butyric acid. Okay, that is 2C. Okay, I got the answer, option number C. I'm teaching you such jugaad tricks, okay? Jugaad tricks I'm teaching you. This is exact Jugaad. Only with 2 options I did the whole match the following. That's why I like match the following. Students will score full 4 marks. They'll not lose any marks, but at the same time some students will be there who will try to do everything correct and then they'll go and make the option wrong. Option number C, yes, perfectly right. Option number C is perfectly right. Okay, that is Aspergillus niger, citric acid, Clostridium butylatum. Okay, that is butyric acid, yeast is ethanol, and this is this. Yes, done. The trigger for activation of toxin of Bacillus thuringiensis. Students, how will this get activated? Okay, the trigger for activation of the toxin Bacillus thuringiensis. This is the reason why when we consume the vegetable, we do not get anything, because we do not have that condition in our stomach. Our stomach is more of, I will not tell the full answer, but yeah, try it. High temperature, alkaline pH of the gut, acidic pH of the stomach, mechanical action of the insect gut. I put Bacillus thuringiensis, that okay, it is affecting cotton plant. I put them, the larvae will consume it and then problem will happen, right? How is that possible? Yes, very good, very good. B is the answer. B for biology, B for beautiful Gopi ma'am, whatever you people are telling. Okay, awesome. The answer is going to be alkaline pH of the gut. That's the reason. For us it does not affect because our pH is acidic. Okay, they will only get activated in an alkaline pH that is seen in larva. Okay, larva. Got it? Yes, very good. Acidity of our stomach does not allow them to get activated. Very good. In 1997, the first transgenic cow, Rosie, produced enhanced quantity of milk, fat-rich milk, human protein enriched milk, vitamin and iron enriched milk. Whenever we are using a transgenic animal, we expect the transgenic animal to be doing something different from the other animals. Yes or no? So here in 1997, the first transgenic cow, that is Rosie. Rosie is the name. Okay, Rosie and Tracy you have to remember. Even if you forget about other people in your life, you have to remember Rosie and Tracy. 2 people very important in your life. Okay, they can give you 4 marks or take your 5 marks. Produced enhanced quantity of milk, fat enriched milk, human protein enriched milk, vitamin and iron enriched milk. Got it? Yes, what's the answer? Yes. Those who joined now haven't marked your attendance. Students, every 5 minutes I have to tell her. Mark your attendance. No. Yes. Very good. Human protein enriched milk. This was the main reason to make this Rosie itself. No, otherwise why will we make it? Normal cows is also giving us fat enriched milk. Right? Yes, Rosie is a cow, Tracy is a sheep, but these 2 people are more important than all the girls in your life. Okay, Tracy and Rosie. Yes. What kind of therapy was given in 1990 to a 4-year-old girl with adenosine DM, that is ADA deficiency? What kind of treatment was given? Gene therapy, chemotherapy, immunotherapy, radiation therapy. So students, this was first ever found itself, right? This was actually a finding, and this finding actually helped because so many things have been, cured right now, right? So this is a very good, it was a first starting, very positive thing. Which kind of therapy was given in 1990 to a 4-year-old kid with ADA deficiency? ADA deficiency. Yes. What's the answer? Yes, A. Ammu ma'am is the answer. Very good. Gene therapy was the therapy given to this 4-year-old kid to cure with ADA deficiency, right? Again, this mechanism, everything I'll teach you. Don't take tension. Next one. A transgenic food crop which may help in solving the problem of night blindness in developing countries. Night blindness. Okay, beta soybean, golden rice, favor slaver tomatoes and starling maize. Students, starling maize and all, don't take tension. Only have doubt with these 2 people. Again, savor flavor tomato also you don't have to take tension. So now I've given you 50-50. A transgenic food crop which help in solving the problem of night blindness in developing countries. Night blindness. Yes, very good, very good, very good. B for biology. B for biology, that is golden rice was given, that is with the carotene, okay, beta carotene, which could help with night blindness, because giving a medicine is difficult, right? So that lacking was rectified, right? Rectified with the vitamin A lack, all that whatever was lacking was rectified by giving golden rice. Okay, very good, fine. BT corn has been made resistant to corn borers disease by the introduction of a gene. Which is that gene? This might be a little difficult for people who do not know the concept, but just give it a try. Is it CRY1AB, CRY2AB, AMPR or TRP? Okay, TRP. BT corn has been made resistant to corn borer disease by introduction of which gene? Which gene? CRY1AB, CRY2AB, AMPR, TRP. Yes, give it a try. Cry only once. Cry only once for this. Don't cry more times. Yes, awesome. Awesome. A for Ames, let's make it, that is CRY1AB is the one that is BT corn has been resistant to. Very good, students. Now we are coming to the last question from my end. Which of the following is a correct match? Crayfish, elasmobranch, cuttlefish, osteo, that is a bony fish, jellyfish, echinodermata, silverfish, arthropoda. Which of the following is a correct match? Correct match. Okay. Crayfish, even if you go wrong in this, it's okay. Let's try finding out. Crayfish, elasmobranch, cuttlefish, your bony fish, jellyfish, echinodermata, silverfish, arthropoda. They're telling which one is right or wrong. Which one is right or wrong. You wouldn't have seen this. Okay, you wouldn't have seen this, but I'll give you. Yes, very good. Yes, very good. Rani, Seep and Krishna, SCP. Okay. Yes, D for doctor. D for doctor. Yes, awesome. The answer is going to be the silverfish, which fall under arthropoda. Students, this is silverfish. How beautiful it looks, no? Yes. Okay, stay back in the stream. Baswa sir will be joining right now. Okay? Yes, Baswa sir is here. Don't run away. Thank you so much, students. Let us know in the comments if the session was helpful, especially my session. Okay? Yeah, if you want more sessions also. If you don't like in comments, students, we are not going to take class. Okay? So, yes, we are going to start with botany. Handing over the mic to the... Please come to the podium, sir.
Thank you so much, ma'am. Thank you so much. Why is it so bright?
Yeah, I don't know. Something went wrong. I'm done with this camera.
Can you reduce the brightness a little?
I made it 4.
Make it 4.5.
Okay, see now?
Yeah, this is better. That was unnecessarily so bright, no? Because I have snips, so many snips. So that won't be visible at all. Yeah. So students, let me know in the chat if you are ready for quick botany questions, and also greenhouse in the chat, and I will know that yes, you are ready for botany questions. Yes. Hello. Good afternoon, everyone. Good evening, in fact. Good evening, everyone. So, well, some stupid. I don't know what is this. Let's have our botany questions. Okay, it doesn't go, is it? Okay. So let me know in the chat quickly if you are ready for botany questions. Yes, I can already see some green hearts as well as certain green leafy things. I think that's a pteridophyte, right? No, that's a proper angiosperm. That's a proper angiosperm leaf, right? So let's start with the first question now. And students, let me tell you, all of these questions are carefully picked at this point so that we are not going to give you extremely difficult questions to discourage you. All of these questions are also not very easy also. So all of these questions are in a right manner. Some of them are PYQs, some of them are most repeated concepts, and they're carefully curated for a specific time right now. Okay, so let's start with the first question from your botany, from your first chapter, that is living world. Living world question. Select the correctly written scientific name. So when you read such scientific name, obviously with your memory you will always go for option number 4. Because you are used to seeing Mangifera indica as an answer in your book also, right? Mangifera indica. But unfortunately, option number 4 is not correct right now. Let's see in the chat how many of you are able to see the answer. I'll give you 5 seconds. I'll give you 5 seconds to answer the question. 5, 4, 3, 2, 1. Time's up. Students, you need to know the scientific name. When you write the scientific name has two parts. Scientific name has two parts. The first part is a genus, then we have species. Genus and species. Genus should always be capital letter, and your species should always have its small letter, and it is always should be italic. You know, it is like Michael Jackson. It is Michael Jackson, right? So it should be always italic. If you're writing in your book, it is supposed to be underline. Underline your book, but since it is bold right here and it's printed, so it should be italic, right? C for chemistry. C for chemistry. C for capto, right? C for capto. Next question. Now, which of the following undergoes meiosis in algae? Now, students, this is a most repeated and my favorite question. It's one of the most repeated and my favorite question. The only way you will be able to solve such questions, if you know the algae cycle, complete cycle. That is, in the algae cycle, first of all, tell me in the chat quickly, what is the main plant body of algae? Is the main body sporophytic or is it gametophytic? First tell me that. Before we go for anything, tell me the main plant body of an algae. Is it a gametophyte or is it sporophyte? Tell me that quickly. You will tell me, sir, the main plant body is haploid in nature. Yes, the main plant body is haploid in nature. That is gametophytic in nature. Gametophytic in nature. Now, students, when the gametophyte produces your gametes, those gametes fuse and they result in zygote. In the case of algae, within that zygote, inside the zygote, meiosis takes place. Inside the zygote, meiosis takes place, which results in the spores. Spores are haploid. Spores develop into a plant body. Which is going to be your gametophytic plant body. Haploid plant body. So where does the meiosis take place, students? The meiosis takes place inside the zygote. We can also call it what? We can also call it zygotic meiosis. So what is the answer? Option number A, B for Baswa sir, C for Capto, D for Diksha ma'am, A for Amazing Gopika ma'am. Is my voice breaking? Always it happens, right? Why does it happen only in my class? Now tell me, is it proper now? Sound check. Is it proper? I have to ask Gopika ma'am, what does she do? Like, does she keep the mic somewhere else? I don't know, in a very weird manner, but is it proper now, students? Yes, it is proper. So obviously answer is going to be inside the zygote, that is zygotic meiosis. Right, zygotic meiosis. Any doubt in that question? Quickly in the chat, tell me. Any doubt in that question? Very easy, right? Very easy, right? Now, next question. Which of the following are colonial green algae? Students, this right here is a NEET PYQ which has been asked so many times. It's a NEET PYQ. The colonial algae. Okay, when I turn, the voice breaks, is it? Okay. I think something is wrong with the mic, or I'm holding it wrong. I have to ask Gopika ma'am for the hacks. Okay. Colonial algae, all of you know, it's going to be the volvox. That is, it is in an apartment. Like a colony they are in, colony. They grow in a colony. They grow inside a colony. Colonial algae, right? Colonial algae. They grow in a complete colony here. Now, quickly tell me in the chat. See, students, I'm going to ask you a lot of questions. Not just the questions which are given here. I'm going to make questions on spot. On spot, I'll make question, I'll ask you. Tell me, which of the following algae is a part of Phaeophyceae? Tell me. Which of the following algae is a part of Phaeophyceae? Students, I want everyone to be super active. I want everyone to be participate. It doesn't matter if your answer is correct, if your answer is wrong. The participation that matters, right? Since you are here, doesn't matter. I will not scold you even if you go wrong. So participation is going to be more important. Now tell me, which of the following is a Phaeophycean algae? That is a brown algae. Brown algae. Quickly in the chat. Brown algae. Brown algae is going to be your fucus. Now tell me, which of the following is a unicellular algae? Quickly in the chat, all of you keep spamming. Which of the following is a unicellular algae? Unicellular algae in this. Obviously, the unicellular algae is going to be Chlamydomonas. Now, which of the following is a filamentous algae? Filamentous algae. Filamentous algae. You'll be like, sir, the filamentous algae is going to be Spirogyra. Clear? Did you understand? So, from one question, you can make so many questions. The limit is only yours. Okay? The limit is only yours. Nice, nice, nice. Next question. Can you go to the next question now? Now, the next question. Which of the following plants produces cones or strobili? Which plant produces cones or strobili? Students, you should understand first of all what is cone and what is strobili. Cone and strobili is nothing but, see, in the case of your pteridophytes, there is something called as, obviously, we have leaves. We have the leaves. But in the case of pteridophytes, we have something called as fertile leaves. Fertile leaves. These fertile leaves have something called as sporangia in it. Sporangia. Okay. And we call this leaf sporophylls. Sporophylls. When many sporophylls come together, we get the strobilus. And there are two examples given you in NCERT. The one example is 𝐂𝐡𝐞𝐤 right here. One example is 𝐂𝐡𝐞𝐤 which is given right here. Quickly tell me in the chat which is the other example. And if you're watching it recorded, quickly smash the like button first of all. Smash that like button. And also I want you to say it loudly even if you're watching it recorded. Doesn't matter if you're not able to type it down. Tell me, which is the other example for cone or strobili other than 𝐂𝐡𝐞𝐤? Other than the 𝐂𝐡𝐞. That is going to be your Selaginella, right? Selaginella as well as 𝐂𝐡𝐞. Nice. Students, all of this should be in your back of your head. See students, the reason we are doing it, all of this now. If you go anywhere on YouTube right now, no one will make you do question practice all the way from starting of a year, right? Students, the counseling has now started. Still, Re-NEET, NEET is still happening. But on this channel, we decided that students should not waste their time, right? And the best way to utilize time is that we take more sessions on the channel. We take more sessions and we'll make sure that you are on point. You're doing assignments, you're doing question practice, you're doing theory, you're doing concepts, everything we're doing on spot, right? Next question, all of you. Next question. The sugarcane plant has dumbbell-shaped guard cells, taproot system, reticulate venation, open vascular bundles. Quickly in the chat answer. Quickly in the chat answer. Yes. First of all, tell me, sugarcane, is it a monocot or dicot? It is a monocot or is it a dicot? Obviously, sugarcane belongs to your monocot. Belongs to monocot. Monocot. Okay. Does the monocot have taproot system? No. Do they have reticulate venation? No. Do they have vascular bundles open? No. They are closed vascular bundles. But do monocots have dumbbell-shaped guard cells? Do they? Do they have dumbbell-shaped guard cells? Dumbbell-shaped guard cells when it comes to your monocots, right? Now tell me quickly in the chat, what is the shape of guard cells when it comes to dicots? Also in the chat, quickly. What is the shape of your guard cells when it comes to dicot? That also tell me in the chat, quickly. Next question. The dark colored fibers present in the nucleoplasm are called as, right? First of all, let me tell you what is nucleoplasm, right? This is your cell right here. This right here is your cell. Cell. Inside the cell, the matrix, right? The matrix, nucleoplasm. So within this nucleoplasm, there is a thread-like structures which are darkly stained. Thread-like structures which are darkly stained, those are called as what? Those are going to be your chromatin. They are going to be your chromatin. Now, can I ask you one more question, all of you? Tell me in the chat quickly. Are you up for one more question from this particular question itself? We will make a question from this question now. What's it? We'll make a question from this question right here. Okay. Tell me, what do you call, which organelle or which structure do you call the factory of ribosomes? The factory of ribosomes. Factory of ribosomes. Where ribosomes are created. Yes, tell me now. Factory of ribosomes. Quickly. Yes. Factory of ribosomes. Factory of your ribosomes. And tell me also, after that, in ribosomes, which type of ribosome do you find in your prokaryotes? Under prokaryotes, do you find 70S or do you find 80S? Under prokaryotes. That is also one of your questions. Easy question, good question. Keep on continuing. Nucleolus, exactly. Nucleolus is called as your factory of ribosomes. Okay. Now, under ribosomes, which one is present in your prokaryotes? It's going to be your 70S ribosome under prokaryotes. Students, if you're able to enjoy the questions, if you're able to solve the questions, quickly mark your attendance by smashing the like button, and also fire in the chat if you're able to solve the questions with high intensity, as well as many questions, as many questions as possible. Right? Quickly fire in the chat. Let's go to the next question now. Next question is oxysomes or the F0, F1 particle, also called as ATP synthase, also called as what? ATP synthase, right? Occurs on. Where does it occur? Where do you find your ATP synthase? Do you find it on outer mitochondrial membrane? Do you find it in mitochondrial matrix or perimitochondrial space that is between the outer and inner layer? Or do you find it in inner mitochondrial membrane? Inner mitochondrial membrane. Now students, such questions are very easy. Such questions can also be asked as where does the electron transport chain takes place, etc. They can also ask you where does the electron transport chain takes place. The answer is going to remain the same. The answer is going to remain the same. That is going to be your inner mitochondrial membrane. Inner mitochondrial membrane. Now, can we make one more question here? Can we make one more question here? Yes, we can make, students. Yes, we can make one more question. Tell me, where does the Krebs cycle takes place? Where does the Krebs cycle takes place? Is it in mitochondrial matrix, inner mitochondria, or is it the cytoplasm? Where does the Krebs cycle takes place? Quickly in the chat, all of you. Students, I am not going to give you breathing space. I will not give you breathing space. The only thing which in my class you're supposed to do is think, answer, think and answer. That is our flow, right? It's going to be mitochondrial matrix for your Krebs cycle. Krebs cycle is going to be your mitochondrial matrix. Okay. Now, under your Krebs cycle, in your Krebs cycle, there is one enzyme which is not found in your matrix. Which is that? That one enzyme which is also present in your electron transport chain. Which is that? Next question, all of you. The centromere splits during the dash stage of meiosis. You can take around 20 seconds, take a breather, think and answer this question. Take a 20-second breather, breathe for some time, think and answer this question now. Think and answer. I'll give you time to think for this question now. I'll give you time to breathe also, drink some water also. 20 seconds, only 20 seconds you get. Succinyl dehydrogenase, yes. Succinyl quickly. The centromere splits. Students, I will tell you this answer right now. I want you to remember this throughout your life. Throughout your life, you should remember this. Okay? Listen to me very carefully now. Listen to me very carefully. This is mitosis here. This is meiosis right here. Meiosis. Under mitosis, you only have anaphase 1. You only have anaphase, that's all. Under meiosis, you have two things. We have anaphase 1 as well as anaphase 2. Because in meiosis, the entire process is happening twice, anaphase 2. Listen to me very carefully. In anaphase 1, we learned it today itself. In anaphase 1, what happens? There is split of centromere. There is split of centromere when it comes to anaphase 1. Splitting of centromere when it comes to anaphase under mitosis. Under mitosis. Now what happens in meiosis? What happens in meiosis? In meiosis, in your anaphase 1, there is separation. One second, students, one second. There is separation of homologous chromosomes. Separation of homologous chromosomes takes place in your anaphase 1. Homologous chromosomes are separating. Anaphase 1 starting stage. Now what happens in your anaphase 2? Anaphase 2. In anaphase 2, there is separation of centromere. There is, or can I say split of centromere. Split of centromere. Okay. Will all of you remember this? Will all of you remember this? Tell me, yes sir, throughout your life I want everyone to remember this, because this is a concept which will keep on repeating, students. They will keep on repeating. So please remember this. Please remember this entire process. Please remember this. Okay? Please remember this now. They will ask you many questions. They'll ask you many questions on this. Many questions will always come. So please remember. Okay? So what is the question now? Question becomes easy peasy, lemon squeezy now. The question says that the centromere splits during the dash stage of meiosis. In meiosis. So in meiosis, where does it split? Centromere is splitting in anaphase 2. Anaphase 2. So what is the answer? Tell me in the chat now. What is going to be the answer? A, B, C, D. Which is the answer? I'm spoon feeding you the answer at this point. Spoon feeding you the answer. It's going to be option number A. Clear? Students, remember, and if you open your book right now, there are many questions on this concept right here. Many questions on this concept right here. So take a screenshot, write it down. Whatever you do, write in your NCERT, write anywhere. Remember it throughout your life right now. Remember it, always remember it. Okay? So, centromere split during the meiosis. Meiosis, split of centromere is anaphase 2. Anaphase 2. Okay? So, one is the answer. What is the answer? Okay? Now, next question, all of you. Next question. Which of the following is not a secondary metabolite? Not a secondary metabolite. Rubber is a secondary metabolite. Ricin is a secondary metabolite. Morphine is also a secondary metabolite. But your aspartic acid right here is the imposter. It's the imposter, which is a amino acid. I don't know what it's doing here. Out of place. Odd one out. Simple odd one out. I don't know why. Okay? Odd one out here. Now, done. Next, glycerol is also called as. Students, always remember, glycerol is a part of your lipid. Glycerol is a part of your lipid. Students, one of the beautiful chapters. Let me know when you want to learn this chapter. Let me know when you want to learn this chapter, biomolecules, and I'll be more than happy to teach this chapter. This is one of my favorite chapters, which is really beautiful. Many concepts to be learned. Many concepts to be learned, right? So, lipids are divided into two categories. One is your glycerol, Other one is your fatty acids. Fatty acids. Glycerol and fatty acids. Now glycerol is also called as your trihydroxypropane. It's written anyway here. Trihydroxypropane. Propane 3 carbon trihydroxy 3 OH groups. Right? A for AIMS. Biomolecules is one of the easiest chapters, trust me. Learn the biomolecules from me once and you will never face any difficulty. You will be able to solve all the questions when it comes to biomolecules. Okay? Don't worry. It's very easy. Next question. Which of the following photosynthesis systems is predominantly found in your stroma lamellae? And I believe we have done this question recently. So you should be able to answer this question with super ease. On stroma lamellae. What is the answer? This is your thylakoids. Between the two thylakoids, the connection is stroma lamellae. This is your stromal lamellae. Right? Stromal lamellae. Now, on this thylakoids right here, on the thylakoids, we have both PS2, PlayStation 1 and 2, right? Photosystem 1 and 2. But on your stromal lamellae, we only find PS1. What is the answer? A for AIMS. Exactly. Easy. Lemon squeezy. Next question. Which of the following is not a product of your light dependent reaction of your photosynthesis? They're asking you which of the following is not found in your light reaction. That is light reaction. Basically in light reaction. Not found. Do you find NADPH in light reaction? Yes. Do you find ATP? Yes. Do you find oxygen is evolved? Yes, because of photolysis of water. But do you find glucose? Do you find glucose in your photosynthesis? Do you find glucose in your light reaction? No. Glucose is produced in your dark reaction. Product of your C3 pathway and as well as your C4 pathway. Glucose is the product of your C3 as well as your C4 pathway. Easy question. Students, students who joined new right now, quickly mark your attendance. I have to tell you repeatedly. Yes, mark your attendance and we will go to the next question now. So do you realize in a span of 20 minutes, I believe we solved half of the questions. Without wasting even a single second, we are like a train, Chikubuku train. We are bullet train, we are going every single question, we are bashing forward. It shows you that how much prepared you are because you are able to answer, right? Next question. So what is the answer? Glucose is the answer, which is not a product of your light reaction. Next question. The absorption peaks of photosystem 1 and photosystem 2 are. This is a very easy question. Laddu question this is. Laddu this is. Laddu. Tirupati Laddu this is. Easy question. Please don't go wrong here. Very easy question. P700 and P800. Photosystem 1, which is around 700 nanometer. Your PS2 is going to be 680 nanometer. Even though PS1 is 1, it is 700. Because why? This was first discovered. Right? It was first discovered. 1 is 700, 3 is 800. Option number D of 4. Next question. Okay, this is again Laddu question. Very easy. I don't know why I keep adding this question. This is a very easy question. What is the correct order of stages of your cellular respiration? What is the correct stage? Cellular respiration. Okay. First is going to be your glycolysis, then it's going to be Krebs cycle, then it's going to be electron transport chain. Answer is option number 3. It doesn't require much of thinking. All of you know this very easily. Glycolysis, Krebs cycle, electron transport system or electron transport chain. Next question. What are the products of your ethyl alcohol fermentation? Very good question and also very easy. Class 10th level question. This is class 10th. What are the products of your ethyl alcohol fermentation? Now I'll ask you more difficult one. I'll ask you more difficult part here. Also tell me in the chat, in this reaction, what are the enzymes involved? What are the enzymes involved? And how much ATP is produced? How much ATP is produced? All three answers I want in the chat now. I want all three answers. You can write comma, 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 answers. Go. Time starts now. Three answers I want. Three answers. Three answers. Not one, not two. Three answers I want. Quickly. Ravindrani, is it lactic acid dehydrogenase? Is it lactic acid dehydrogenase? Lactic acid dehydrogenase is production of lactic acid, right? The product here is going to be what? The product here is going to be ethanol. Exactly, Sangeeta. Exactly, Sangeeta. The one enzyme is alcohol dehydrogenase. One enzyme is alcohol dehydrogenase. Along with that, there is one more enzyme. No, one more enzyme is there. I believe there are two enzymes here. Other one is pyruvic acid decarboxylase. You don't remember, no? Remember, how many ATP? 2 ATPs are produced. Only 2 ATP is produced. 2 ATP. Enzymes, there are two enzymes. That is alcohol dehydrogenase, pyruvic acid decarboxylase. Mistakes happen because you don't revise. Keep on revising, mistakes will not happen. Okay? See, that is why we are making the sessions. Why do you think we are making the sessions, students? Why do you think we are making the sessions? We want you to make your mistakes from the early stages. Like, I know, see, do you remember when your NEET 2024 was very close by? When it was very close by, how many questions did we do? Every single day we should do mock test. Every single day. But that made us realize that our YouTube students are still lazy. Throughout the year they might not work. Throughout the year they will not work. So we realized that it doesn't matter if anyone is doing it or not. For our students, we will do it from the day one. We'll do it from our day one question practice. Now, acceptor of your acetyl-CoA in Krebs cycle is acceptor of acetyl-CoA in your Krebs cycle. That is going to be your oxaloacetate. OAA plus your acetyl-CoA. Acetyl-CoA will produce your citrate. Now tell me in the chat, citrate is how many carbons? Citrate is how many carbon? How many carbon is citrate? What is the carbon number here? What is the carbon number for citrate? What is the carbon number for oxaloacetate? What is the carbon number for acetyl-CoA? Quickly in the chat. Quickly in the chat, all of you. I'll ask you one more question after that. So, your citrate is 6 carbon. Okay. Citrate is going to be 6 carbon. Okay. 6 carbon. Now, students, this 6 carbon becomes 5 carbon at one point. This 6 carbon becomes 5 carbon. Now, tell me, which is that 5 carbon molecule? Tell me. Which is that 5 carbon molecule under your Krebs cycle? Think. All of you. Which is that 5 carbon molecule? I'll give you a clue. It starts from alpha. It's going to be your alpha ketoglutarate is 5 carbon molecule, and that is a PYQ. That is an important question. You should know which carbon. Okay. Then after that we have a 4 carbon molecule. The 4 carbon continues till all the way till oxyl acetate, 4 carbon. Your acetyl CoA is 2 carbon. 2 carbon. 2 plus 4 is going to be your 6 carbon here. Again, 6 carbon is citrate. Everything is in balance. Everything is in balance. Okay. Nice, nice, nice. You're able to answer. Next question. Heterophilous development due to the environment can be an example of senescence, plasticity, dormancy, none of the above. First of all, tell me, what is heterophily? Heterophily means different types of leaf. Different types of leaves. Different type of leaves. Okay. Heterophily can happen because of two main reasons. That is, one can be the phase of the plant cycle. Phase of plant. That is, a leaf can be different in your juvenile stage. Leaf can be different in adult stage. Quickly in the chat, examples for this. Examples for this. Other one is based on environment. Other one is based on environment. That is, in aquatic, the leaf will look something like this. Aquatic. But in terrestrial, leaf will look something different. Leaf will look different in terrestrial. Example for that also I want. Example for this also I want. Examples here. And this is called as plasticity. It is called as plasticity. Plasticity. Answer in the chat quickly. For examples for your phase of a plant, that is going to be cotton, coriander and larkspur. Cotton, coriander and larkspur. And the example for environmental or environment is going to be your buttercup. It's going to be buttercup. Students, every example is important. Biology is a subject where you need to keep revising. Biology can give you the full marks or it can take away the marks. It all depends on your hard work. If you are able to remember the examples, if you are able to remember the reactions, if you are able to remember the different structures, diagrams, flowcharts, marks are yours. Marks are yours. Next question. Dash is used to speed up the malting process in your brewing industry. Malting process in your brewing industry. Students, remember, your biology demands extra work. Biology demands extra hard work. So, you do your hard work right now. You remember, recently we did this. It's going to be gibberellic acid. It's going to be your gibberellic acid, Ga3. Ga3. Right? Malting process. A type of population interaction between the two populations of two different species where one of them is benefited and the other one is neither benefited nor it is losing anything. One is going to be benefited, other one is not benefited or not losing anything. That's going to be your commensalism. It's going to be commensalism. Nice. Commensalism. It's going to be commensalism. Okay. Next question. Dash proposed the Ribet-Popper hypothesis. Who gave the Ribet hypothesis? Popper hypothesis. Who was a scientist? Who told us that the ecosystem, the entire ecosystem is like an aeroplane. Entire ecosystem is like an aeroplane, and in aeroplane we have nuts and bolts. Nuts and bolts. Similarly, in our ecosystem we have different animals. Remove one nut, nothing will happen. Remove one animal in the starting, nothing will happen. But to remove all the bolts, remove all the animals, ecosystem will also collapse. Your aeroplane also will crash. Now imagine you remove one nut or a bolt or a screw of the window. Window opens. If the window or the door opens, the plane will immediately crash. Similarly, in your ecosystem, there is something called as endemic species, right? Endemic, not endemic as well as your what is the name? What is the other name of that? Endemic as well as your ecological significant species. Students, what do you call the most ecological species? Like a banyan tree, elephants, banyan tree and elephants. What do you call them? The ecologically most important species. Ecologically most important species, if you remove them, the entire system will collapse. Not endemic species, I'm talking about species which you remove, the entire system will collapse. For example, if you remove the banyan tree, the entire system will collapse. Not endangered, not endangered, not endangered species. You remove that species, entire system will collapse. Not extinct, not extinct. Keystone species, exactly Krishna, keystone species. If you remove that species, entire system will collapse. For example, if you remove the elephant, entire species, entire ecosystem will go. Remove the banyan tree, entire ecosystem will collapse. They are the most important species in the entire ecosystem. This was easy question. Paul and Rich. Next question. Which of the following best describes the endemism? What is endemism? Quickly go. What is going to be endemism? Co-extinct species? No, not co-extinct species. Which of the following best describes the endemism? Species confined to an area and not found anywhere else? Obviously, that is the answer. Species, endemic species are species which you will not find anywhere. For example, penguins, you will not find penguins randomly anywhere. Polar bears, you will not find polar bears in your Western Ghats, you will not find polar bears in your African jungles or Amazon forest. Polar bears will always be in Arctic regions, unless you keep them in zoo, but that is their main habitat. How many cells, it's the most easiest question for today, most easiest question. How many cells and nuclei are present in a mature embryo sac? In a mature embryo sac, how many cells and how many nuclei is there? How many cells and how many nuclei is there in a mature embryo sac? In a mature embryo sac, there are 3 cells here, 3 cells here, central cell, tell me quickly in the chat what is the answer. 3 antipodals, 2 synergids, 1 egg cell and 1 central cell. 7 celled and 8 nuclei. 1, 2, 3, 4, 5, 6, 7 celled. 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei. Option number D. Next question. The study of Bakane disease of rice led to the discovery of. The Bakane disease, the foolish disease. The foolish disease led to the discovery of. The foolish disease led to the discovery of. Where a rice plant, students, a rice plant grew so tall. The rice plant grew so tall that it is not producing any seeds. It is not producing any seeds at all. So tall it became. Bakane disease. That gave rise to the discovery of your gibberellins. Gibberellins. Nice question. Now, next question. Identify the basic amino acid from the following. Which of the following is the basic amino acid? Basic amino acid. Basic amino acid. Which is the basic amino acid? It's going to be your lysine. It's going to be your Lysine. Lysine is the basic amino acid. Now next question. Inulin is a polymer of. Inulin is a polymer of. Inulin is a polymer of. Now not glutamic acid. Glutamic acid says acid. How can it be basic? It's going to be lysine. Lysine and arginine. Lysine and arginine. Oh, you're telling this answer, is it? Previous answer you're telling me. That is one. That is one. That is one. Basic amino acid done. Now, inulin is a polymer of. Students, biomolecules question. Deliberately, I have added more questions of biomolecules because I know some of you are keeping your chapter as a backlog. Tell me honestly in the chat, how many of you think biomolecules is the major backlog for all of you? Tell me. Is it a backlog chapter for you? Backlog chapter? Biomolecules? Tell me. So, inulin is a polymer of fructose. Long chain of fructose. Long chain of fructose. Inulin. It's not insulin, it is inulin. Read properly. Insulin. Now, ABO blood grouping is an example of. ABO blood grouping is an example of. Quickly in the chat, answer. ABO blood grouping. See, in ABO blood grouping, we have two dominant alleles. Dominant alleles, one recessive allele. So, two dominant alleles are there. That is an example of. That is an example of. Quickly in the chat, all of you. That is an example of multiple allelism. Nice. Nice. Multiple allelism. Which of the following is not an autosomal recessive disease? Not an autosomal recessive disease. Not an autosomal recessive disease. Quickly in the chat, you have 10 more seconds. I'll give you a quick example. Hello, sir? 5 minutes. That'll be done. Is not a autosomal disease. Students, I know some of you might be going wrong because you do not remember this, but remember your hemophilia is a X-linked recessive disease. X-linked recessive disease is your hemophilia. So it is not an autosomal disease, it is X-linked recessive disease. There is no way you can do this, you have to remember this also, you have to remember this. Next question. The phenomena where a single gene exhibits multiple phenotypic expression is called as a single gene, many expression. Single gene, many expression. Single gene, many way expressions. That's going to be your pleiotropy. Nice. Pleiotropy. Pleiotropy. So one gene, multiple expression. Like your mother in the house. Your mother in the house is able to do all the work, right? She can do anything in the world. Mother is cooking, cleaning, making your food, making you ready for the school. She's sending you to school, sending you to college. After she come back, she's taking care of you. She's also working. So mother is doing all the work. So pleiotropy is like your mother. Okay, pleiotropy is the answer. Look at this question now. These are regarded as the major cause of biodiversity loss. Right? There are totally something called as evil quadrate. There is something called as evil quadrate. There are 4 of them. The first one is called as the most destructive and the most important one is going to be your habitat loss and fragmentation. Habitat loss and fragmentation. Then we have overexploitation. Then we also have your co-extinction. So 3 are given here. 3 are given. The only thing which is missing here is alien species invasion. Alien species invasion is missing here. Now tell me, what is the answer then? Major cause of biodiversity loss. A, B, D. A, B, D, yes. Yes. Option number C. Option number C. Okay. Look at this question now. Match the following. Robert May told about your global species diversity about 7 million. Alexander von Bolt, who gave the species area relationship. Paul Ehrlich, who gave the river property hypothesis. David Tillman, who gave the long-term ecosystem experiments using outdoor plots. What is the answer? What is the answer? And students, this is the last question for today. Last question for today. Last question for today. A is 3, so A is going to be 3. Okay. So these two are gone. And B is 1. B is 1 here. B is 1 is here. Done. Done. Students, you actually solved in the less than, I don't know how much of time I took. You solved more than 35 questions which is on the board. You solved 35 questions which are on the board. Now imagine how many questions you solved when I was even explaining. Like, you solved many questions which are on the board. I asked you around 10 to 15 questions I would have asked. So in total of such a less time, you were able to revise so many concepts. So many concepts you were able to revise. And you were able to answer them. Your confidence is brimming. And you also could not able to answer some. So I feel like some of you went wrong, right? Some of you went wrong here and there. Some of you went wrong here and there. You were able to tell. Now next time if the same questions come, will you make the same mistake? No, you will not make the same mistake. That is the beauty of question solving. That is the beauty of question solving, because when you solve the questions, multiple area gets revised. You will also know where you're confident. You will also know where you're making mistakes, so that you do not repeat again. And if you do this throughout the year, imagine your confidence at the end of the year, students, at the end of the year. So if you're not subscribed to the channel, quickly subscribe to the channel. Yes. And do that and like the video. Let us know in the comment section if this session was useful to you, because Shreya sir is going to come now. Do not go anywhere. Wait. Don't go anywhere. Shreya sir is coming now with physics questions. And you know physics, how important physics is going to be. Right. So Shreya sir is coming now. Let's invite Shreya sir. Obviously not come here. Sir is doing from his house. And I will scoot now. Yes. Let us know in the comment section how was this class, right? Let us know in the comment section how was this class. And before we go, let me tell you something. Before I go, let me tell you something. I don't know why this is happening. Students, if you are not a part of DNA batch still, it doesn't matter if you're going to offline, you're going anywhere. This is the batch for you, because in DNA batch, we do things in a much more detailed way, intricate way. You will have weekly quizzes. You have alternate weeks. You will also have alternate weeks, weekly quizzes. Then you have your Tatva books. We'll be giving you a PDF copy. We'll also be doing quizzes in the class. It's much more interactive than this. It's a two-way class. We have multiple teachers two teachers in one class to solve your doubts. So if you're not a part of DNA batch, quickly join right now. Link is there right here. See, right here. NEET 2025 DNA batch, where we are doing question practice every single day almost. Right? So that is your DNA batch. And also let me tell you, tomorrow I'll be taking a zoology class. That's right. Tomorrow I'll be taking a zoology class, that is your human reproduction, exactly at your 4 p.m. Exactly 4 p.m. we'll be starting with human reproduction. Let me know in the chat if you're excited for that also. And quickly go and smash the like button. First of all of you, I want to see this like button at least 50 before I start tomorrow's class. 50 likes here. It should be here. Because let me tell you, we'll be doing theory plus PYQs. Theory plus PYQ plus NCERT lines. So you do not miss out any single point. Any single point you do not miss out. So with that being said, let's welcome Shreya sir. I'll call Shreya sir right now. And let's welcome Shreya sir now.
just for your sake. And you will also see that we are giving you lots and lots of free content as well. As you're watching it, in the description box you will see that there is this treasure box which you have. I'm not sure if it is here right now, somewhere it is. Maybe in the some other video. Yeah, so where we are putting up all the PDFs, all the lectures, all the DPPs, everything in a very systematic manner, including the NCERT questions. Okay, so get set started, guys. It's time to begin. And I want every student to mark their attendance right over here quickly. Make this go up above 100 right now. Not just that, I want you to be completely active in the chat box as well, in the terms of the answers and your enthusiasm. Hello, Suhasini, botany lecture going to take zoology. What do you mean? Botany and zoology is just over. Yes, Baswa sir was taking botany. Yep. Okay. So Banot, welcome. Hello, Naina. Naina has come out of the box. Okay. Hello, Krishna. Warrior Pandu. Hello, Pavitra. Happy evening. Happy evening, Krishna. Okay. It's time to begin the class. And today we are going to do lots of questions on basic maths, which has a lot of mathematical aspects. That's the basics part. And obviously the concepts of vectors and scalars will solve as many questions as possible in today's class. Okay, so get ready for it. All right, so let's start with some questions, my dear students. Let's start with some questions, my dear students. Here is the first question coming up on your screen. Example: if it is given that the maximum upon the minimum ratio of the resultant of two vectors is 9:5. Question is, find the ratio of the vectors. Find the ratio of the vectors magnitude. That is the question. Okay, let's see how do we solve them. First of all, the maximum to minimum ratio for the resultant of two vectors will happen in the following two cases. First, if the two vectors which are being added are perfectly aligned versus the two vectors are facing opposite to each other, in this case, the resultant which is going to be produced is going to be maximum, having the magnitude a plus b. If they are opposite, the resultant is going to be minimum, having the ratio, having the magnitude a minus b. So this is minimum and this is maximum. It's clearly mentioned a plus b divided by a minus b is 9 divided by 5 is nothing but 9 divided by 5. Now, how do I find a and b separately? I feel I can use componendo dividendo ratio. Exactly. Remember that componendo dividendo property? Whenever I see plus and minus on the top and the bottom, I think of componendo dividendo. You take the numerator, add to the denominator. Take the numerator, subtract it with the denominator. Same thing, do it on the RHS. So it will be a plus b, which is the numerator. Add it to the denominator, which is a minus b. Subtract it with the denominator, which is a minus b. Do the same thing on the right hand side. So it will be 9 plus 5 divided by 9 minus 5. What will this be? B and B cancels, it will become just 2 times of A. Here, A and A will cancel, B will get added with B, so it will become 2 times of B, which will be 9 plus 5, which is nothing but 14, 9 minus 5, which is basically 4, 2 cancels, so it will just become A divided by B. 14 and 4 both divisible by 2, hence I can just write 14 divided by 2 is 7, 4 divided by 2 is basically 2. Hence the ratio is 7:2. How many of you wrote 7 by 2? Very good, Krishna. Very good, Harborine. Very good, Madhumita. Awesome. I want all your spams coming out over there. Awesome, Pavitra. Very good. Kudos to all of you. You guys have given the correct answer. Moving on to the next question, which is going to come up on your screen. Here it is. Here it is. Next question is over here. Find the value Okay, find the value of root of 1005. Find the value of the root of 1000 and sorry, 10,005. 10,005. Okay, approximately how much will it be? This is also a very interesting question. The moment you see such questions, you should remind yourself of binomial approximation. Binomial approximation. So how do you write it in the binomial form? And what was that binomial approximation? Those of you don't remember the formula, let me write it down for you. 1 plus x whole raised to n is, this could be minus also, 1 plus minus nx, assuming x is much smaller than this number 1. That was your binomial approximation formula. So let's use it. And root means it is nothing but raised to half power. So I can just write this as, 10,000, okay, 10,000 plus the number 5, 10,000 plus the number 5 whole raised to the power half. Now let me take 10,000 common. Why did I do plus 5? You will see it in the next step. It is a beautiful manipulation. Let me take out 10,000 common raised to half. So what is left inside is 1 plus 5 divided by over here 10,000, and this whole thing raised to half. I just taken out 10,000 common outside, so 1 is remaining here, and since 10,000 was not there, so I just put it in the denominator. That's all. Now what is the 10,000 power half answer? It's the square root of 10,000, which is nothing but 100. 100 into 100 is 10,000. So the square root of 10,000 is nothing but 100. And this now looks to me like this formula. So I might as well use the binomial approximation. So now this is no longer equal to, this is approximation. 1 plus, this is like x, this is like n, this is much smaller than 1, which is true. N is nothing but half, x is nothing but 5 divided by nothing but 10,000. Okay, perfect. So this will be approximately 100 into 1 plus. Now see guys, things will get cancelled off a little bit here and there. You can see that. Okay, so this 5 will go with this 10,000 with 2,000, and this 2,000 into 2 will become 4,000, right? This will just become 4,000. So you can just write it by 1 by 4,000 over here, and this will now become 100 plus 100 divided by your 4,000. This 0, this 0, this 0, this 0 gets cancelled. You just have 1 by 40. 1 by 40 is nothing but 0.025, 0.025. So hence this will be nothing but 100 plus 0.025, because 1 by 4 is 0.25. So 1 by 40 will be 0.025. So hence the answer will be 100.025. How many of you wrote that answer? Oh, don't say approx 100. That will be wrong. Because if it was 100, then what was the whole point of this? I wanted that small difference, because sometimes what happens in some questions, you will be asked the value of what is the root of, I mean, it will come as a calculation step. This minus 100 is the final answer. Then you cannot say this is 100, then 100 minus 100 will be 0. So we got just now that this answer was nothing but 100.025, this minus 100. So hence the approximate answer will be 0.025. Maybe it is used somewhere. Maybe you are just trying to find, example, the sign of this. Maybe you're trying to find the sign of this. So essentially you are trying to find the sign of this number is 100.025 minus 100, and then this will just become sine of 0.025, and the sine of a small angle is that angle itself. So it will be again 0.025. Here I have used our small angle approximation. Small angle approximation. Is that right? That is what I have done. Got it? So that is why sometimes You might have to use binomial as a tool for simplifying the calculation. Nobody in the NEET exam will ask you this question. It will be a step in one of the bigger problems or one of the calculative problems. Understand, it is just to ease your calculations, nothing more, nothing less. Going ahead to the next question coming up on your screen. Okay, get ready for this. The next example is as follows. Find the simplification of log 20, let's say log 24 to the base 2. Log 24 to the base 2 in terms of the smaller logs. In terms of the smaller logs, like in terms of log 2 or log 3, something like that. In terms of basically log 2 and log 3. How can you write it in terms of those smaller logs is the real question. Come on, my dear warriors. What can you do with 24? You can factorize it. 6 4s are 24. Okay, 6 4s are 24. So just use that. Come on. So 6 4s are 24. 6, can you write it in terms of something? Okay, or you can write it as 8 3s are 24. So try to do those manipulations and see what do you basically get. See what do you basically get. Come on, my dear students. See what is it that you are getting. See what is it that you are getting. Come on, come on, come on. Yes, see what is it that you are getting. And by the way, let me also remind you of the log formulas. ln2 is approximately and ln3 is approximately. ln3 is approximately 1.09 and this is basically 0.67 approximately, right? So these are your approximate formulas which you should know. Can you write it in terms of some simpler logs eventually and see what the answer is going to be? Yeah, come on. Yeah, think about it. 0.6, sorry, this is 0.69, not 0.67. This is 0.69. Yes, come on, come on, come on, my dear students. 24 can be written down as, 24 can be written down as, 8 into 3, that will be better. And 8 itself is 2 cube. So I'll just write it like this. And this whole thing to the base 2. I do not like it to the base 2 because when I say ln, ln is to the base e. So first thing first, I have to also convert it. Yes, first thing I have to convert it. Okay, very good. So how do you convert it? Come on, Akira, Srishti, Harborine, Naina, Pavitra, Krishna, everybody think I'll have to get rid of this too. Use our chain rule. So how does our chain rule work? It will work something like this. Let's write it as 2 cube into 3 to the base e. The whole thing into log of e, the whole thing to the base 2. This is perfectly fine. Chain rule, the number to this base, number to that required base, into that required base to that original base which you wanted. This is your chain rule. Now, whenever you have this, you can also write this as, whenever you have this, by the way, you can also write this as, shift the base to here and shift this to the base. So this is basically log of 2 to the base e. Over here, you will see this is nothing but 2 cube, and whenever you have multiplication, you can convert it into addition. So hence, I can also write it as log of 3 to the base e plus log of 2 cube to the base e. But wait a minute, I know what to do. Whenever you have a power inside the logarithm, that the number comes outside. So it will be 3 times of log 2 to the base e plus log of 3 to the base e, the whole thing divided by log of 2 to the base e. Amazing thing, log of 2 to the base e can get cancelled in the first part. The second part it will remain. Hence that answer will be nothing but 3 plus ln 3 divided by ln 2. Just check this out. Log 2 initially will get cancelled out in that first term. In that second term, log 3 is there, log 2 is there, numerator, denominator. Hence, this is the answer in the simplest form. If you want to substitute the values, yes, you are free to do so. You can put it as 1.09 divided by 0.69. Okay, you can find out the exact value. But this is how you can simplify it from here to here when the base was only something else. Excellent. Understood how to use addition, subtraction rule, change of base rule, chain rule, so many things. Reciprocal rule was also involved over here. Going to the next question coming up on your screen. Find the value of sine of 75 degrees. Also find the cos of 15 degrees. Okay, using any subtraction, addition, whatever rules you have learned in trigonometry. It's completely up to you. It's completely up to you. Okay, how you want to do this. Okay, how do you find sine of 75 degrees? Can you write 75 as the sum or subtraction of two things? I think so. You can write it as sine of 45 degrees plus 30 degrees. Perfect. So this is sine of a plus b formula, which means you have to write sine 45 cos 30. Then if there is plus, you will put plus for sine. For sine, sine is same. Then you will put cos 45 into sine of 30. Very nice. What is sine of 45? It is 1 by root 2. Cos of 30 is root 3 by 2. Plus cos 45 is 1 by root 2. Sine 30 is 1 by 2. 2 root 2 is the same everywhere. So 2 root 2 is the same everywhere. And I have 1 plus root 3. So that is sine of 75 degrees. There you go. Let's do the next part of the problem. Cos of 15. Can I write 15 as something plus something, not plus, minus for sure. I can write it as cos of 45 degrees minus 30 degrees. This is perfectly fine. Guess what? You don't even have to do this. You don't even have to do this complete thing. Sine 75 will anyways be cos 15. Just like sine 30 is cos 60. Sine 37 is cos 53. For complementary angles, what is same? The sine and cos will be the same. So sine of an angle is the cos of the complementary angle. Cos of an angle is the sine of the complementary angle. Even if you go do this by the method, you will still get it as 1 plus root 3 divided by 2 root 2. You don't have to really solve it. Okay, I hope this is absolutely clear. I hope this is absolutely clear. Okay, all right, great, awesome. Akira, unless and until you have been living under a rock, probably you do not remember what and all problems have happened. It's not like there were no problems. The problems are still there. It's just that many people are choosing to overlook the problem. It's not like NEET 2024 did not have a problem. Obviously, you should be blind or you should be deaf to not know that there were problems. Okay, so unless and until you are living under a rock, choosing to ignore, maybe the brain did not develop in you, maybe then you can say so. Yeah, cool. So moving on to the next question coming up on your screen, and here it is. So let's say we want to solve this particular problem. I want to find what is 2 plus tan theta upon 2 minus tan theta, which is equal to let's say 4 divided by 3. Okay, I want to find the value of tan theta over here in this particular question. I want to find the value of tan theta in this particular question. What do we do? Very simple, guys. There is plus, there is minus. I can see similar terms over here. Okay, and let's say this was over here, maybe cos theta also is there over here. Oh no, not needed. It's not needed. Okay, so now I'll just use component to dividendo. So guys, 2 plus tan theta upon 2 plus tan theta. I'll just put it as it is. This is just the numerator. Add the denominator here, subtract the denominator. Here it will be 2 minus tan theta, and here it will be nothing but 2 minus tan theta again. Here it will be 4 plus 3 upon 4 minus 3. What will happen over here? Tan theta will cancel. 2 plus 2 is basically the number 4. Here what will happen? 2 and 2 will subtract. Minus will become plus. So it will become 2 times of tan theta. This will be nothing but 7 divided by 1. 4 goes with 2 times. So taking 2 over here, taking 7 below over here, 2 by 7 is nothing but tan theta. Therefore, theta will be nothing but tan inverse of 2 by 7. Tan inverse of nothing but 2 by 7. That should be the answer. Yep. Awesome. Kudos to those of you got it till here. Next question coming up on your screen. Here it is. Find the maximum and the minimum value of 5 minus 2 cos theta. 5 minus 2 cos theta. What is the maximum value of that? That is the real question. So for solving such questions, I'll tell you a simple methodical way. You will not get confused at all. Probably this could be a part of a big question. Maybe the force came out to be 5 minus 2 cos theta. What is the maximum force? What is the minimum force? The velocity is this. What's the maximum velocity? What's the minimum velocity? So such type of questions, how do you solve? See, I know cos is always going to be between -1 and +1. That is for sure. So what will I do? I can see the variable over here is theta, 5, 2, etc. are constants. So cos of theta will be always less than 1 and it will be always more than 1, -1. Okay? Okay. Great. So Akira, nobody is asking you to watch here. Please get lost. Why are you wasting your time here? And we didn't waste anybody's time. If standing for the students, according to you, was poor performance, then so be it. I will again and again make sure that I do this poor performance for the students. If I have to stand for the students, if so-called students like you have a problem with it, I have no issue. I'm going to just overlook students just like you or people just like you. Every time there is an injustice, I will fight for the students. Every time something has gone wrong, we'll speak for it. Because people like you who are cheaters probably, who probably might have bought the papers, are feeling sad that these issues have come up, probably because of that. So really, we stand over here for honest students. We understand the hard work which students have put in over here, and when something has gone wrong, their hard work has gone wasted and people with money have won. Okay, if paper leak, all these things, which has happened at many centers. It's not like it has not happened. CBI and so many people have been arrested. CBI has arrested so many students and so many masterminds throughout the country. Bihar paper leak happened. Gujarat paper leak happened. Paper was not given on time. Paper was snatched away. Invigilators wasted the time. Students in Tuticorin got different papers. Students in so many northeastern centers, they did not get the complete paper timing. If speaking for them, according to you, is waste of time, I think you do not even deserve to be a NEET student. Maybe you should go study something else. You really don't understand what a doctor means. You really don't understand what it means to study for NEET. You don't understand what it means when a student studies for 5 years, 4 years, taking multiple drops, persuading their parents, asking their parents to, give them money or give them support for their drops. You really don't appreciate that. That is the reason why you're speaking like this. So make sure you think about it, what you're speaking, and then come over here, or else don't waste our time, because there are students who want to prepare for NEET 25, which is going to be more competitive. So honestly, we don't care what you think. Okay? Right. So moving on to the next one. What do we do now? I can see that it is being multiplied by the number 2. Cos theta is being multiplied by the number 2. So I'll multiply everything by the number 2. Everything I'm going to be multiplied by the number 2. And by the way, Akira, you are wasting our time. We are going to block you. By the way, let me tell you, any student who spams like this, they just get blocked in the NEET channel. Okay? So simply don't waste your time. They're just blocking you. Okay. Now, so 2 times. So this will be nothing but 2 times of cos theta, and this will be also multiplied 2 times, and this will be also multiplied 2 times, so -2. Oh, sorry. Yeah, correct. Now, what I can do over here to get this 5 minus 2 cos theta? I can say 2 cos theta is there, but I want to get 5 also. But in fact, there is -2 cos theta, so I'll multiply it with -1. Now, remember this simple thing, guys. Whenever, say for example, x is less than y, if you put minus x over here and minus y over here, this inequality changes. The inequality will change the moment you go to negative sign. Yes, keep that in mind. The inequality will go to exactly the opposite side when you multiply it with a negative number. So what you will do over here to get minus 2 cos theta, minus 2 cos theta, this will become more than equal to, this 2 will become minus 2 and this will again become plus 2. That's it. That's the major change. Now I will add the number 5 over here. So 5 minus 2 cos theta more than equal to, add 5 over here, also add 5 over here. 5 plus 2 is 7 is more than equal to 5 minus 2 cos theta more than equal to 3 over here. So what does this tell you? 3 is the least value, so this is the minimum value, and 7 is the maximum value. So the maximum value of this is 7 and the least value of this expression is the number 3. That's all you need to do. I hope this is clear. So these kind of questions are also very common. You might have seen this. Okay. Another type of question which I have seen probably in the chapter of friction, which is also very interesting. So imagine the question goes like this. There is a force which is, let's say, some number A or say the number C divided by A cos theta plus B sin theta. And then they ask you, find the basically maximum and minimum value of that f. Find the maximum and the minimum value of f. How do we do this question? Now let me tell you, when there is just sin theta and cos theta, it is very simple alone. But when they are combined together, there is a simple formula which you have to remember. What is that simple formula? Let me remind you of it. It is nothing but A cos theta plus B sin theta is always less than or equal to the root of their coefficients, which is A and B, squares and added. That means A square plus B square. And this is less than or equal to, root of A square plus B square minus sign. That's it. It's more than this and it is less than this. Probably we can have some numbers also written down over here. We can put some random numbers for, making it look like a or feel like a problem. I can put some numbers like 3, maybe this is 10, or maybe this is 4. I just put some random numbers over here. So if this formula were to be true, this expression here, I can see 3 cos theta plus 4 sin theta is just like this. So I can say 3 cos theta plus 4 sin theta is less than or equal to root of 3 square plus 4 square, and this is more than or equal to root of 3 square plus 4 square with a negative sign. 3 square is 9, 4 square is 16, 25's root is 5. So minus 5 less than equal to 3 cos theta plus 4 sin theta less than or equal to, this is also plus 5. But now you can see this whole number is in the numerator, but here it is in the denominator. So I will just take the reciprocal of this. I'll just basically take the reciprocal of this. Now when I take reciprocal, what do you think happens? Let me again tell you this simple thing. If x is more than y, then 1 by x and 1 by y, what will be the comparison? Will it be still more than? Will it be still more than, guys? Think about it. No, it will be now less than. So when you take reciprocal, also the sign will flip. The sign will again flip. So keep this in mind. So hence, when I take the reciprocal of this thing, which is 1 by 3 cos theta plus 4 sin theta is going to be more than or equal to, this will be 1 by 5 and here it will be minus 1 by 5. But wait a minute, in the numerator I want the number 10. No issues. I'll put the number 10 with this whole thing, which is 3 cos plus 4 sin. This will be more than or equal to 10 by 5 and here it will be nothing but minus 10 by 5. Minus 10 by 5 and plus 10 by 5, guys, What is this 10 by 5? It is 2. And what is this negative 10 by 5? It is nothing but minus 2. So minus 2 is the minimum value and the maximum value is basically plus 2. That's how you find it out. That's how you find it out. Okay, I hope this is very, very clear. I hope this is very, very clear, my dear students. Very nice. Let's move on. Let's move on to the next question, which is going to come up on your screen. And here it is. Question is, find the value of sine. Find the value of sine 0.7 degrees. Find the value of sine 0.7 degrees. How much is this sine 0.7 degrees going to be? Okay, let's try to figure this out. What do you do in this particular case? Think about it. What do you do in this particular case? Think about it. It's in degrees. It's a small angle. Maybe I need to use small angle approximation. But keep one thing in mind. Sine of theta is roughly theta if theta is in radian only. So, if it is in degree, how do I convert it into radian? So, if theta is in degree, you multiply it by pi and divide it by 180. Divided by 180, what you get is theta in radians. Theta in basically radians. So, first of all, the step should be to convert these degrees into radians. Only then I can use the small angle formula. So, sine of 0.7 degree, I can write it as 0.7 into pi, the whole thing divided by 180 radian. This is perfect. And how much is this going to be? Well, this is a very small number, and whenever the theta is small, I can write the sine of that angle to be the theta only. So it will be nothing but 0.7 multiplied by pi, the whole thing divided by 180. If you want, you can simplify this. This is, by the way, approximate. This is not exact. So I can write 0.7 as 7 by 10. Pi, I can write it as 22 by 7 and 180 are just over here. So you can see 7 cancels, 0 0 adds up, 22 goes with 2 11 times, 18 goes with 2 9 times. So this will be nothing but 11 divided by 7 7 has got cancelled, my bad. 18 divided by 2 is 9. 0 0 adds up to become 100. So that's it. It's just going to be nothing but 11 divided by 900. That is the answer. 11 divided by 900. If you want to convert it into decimals, that also you can do. Is that okay, my dear students? Is that okay, my dear students? Understood? Clear? Can we go ahead? Very good. Let's move on. So this is again the application of small angle approximation. Let's do the next question coming up on your screen. Find the derivative with respect to theta of sine square theta. Find the derivative of sine square theta. That is the question. Also, on the other side, the question is find the derivative of sine of theta square. Find the derivative of sine of theta square with respect to theta. Two different kinds of questions. Probably we need different approaches also. Probably we need different approaches also. How do we do this? I'll give a hint. Maybe we need chain rule. Maybe we need chain rule. Maybe we need chain rule. So what could be the chain rule? Think about it. Sine of theta square. Guys, derivative of sine x with respect to x. Derivative of sine x with respect to x is cos x. But derivative of sine of x square with respect to x, that is not cos of x. So I need to ensure that whatever is there inside the sign is matching over here in the denominator. Only then it will be correct. Otherwise, it will not be correct. Otherwise, it will not be correct. Okay. So what do we do? What do we do over here? How about writing it as derivative of sine of theta square with respect to theta square, and put over here theta square and then put this original d theta over here. Now it looks like something. Whatever is there inside the bracket is also there over here. I've just used the chain rule. Perfect. So now derivative of sine of x with respect to the same number is cos of x. Hence, this will be cos of theta square, the first answer. This part over here is like saying, this part over here is just like saying that x raised to n formula. It is nx raised to n minus 1. Exactly. So n over here is 2, x over here is theta, so 2 minus 1. 2 minus 1 is just 1, theta raised to 1 is just theta. So hence the answer, oops, this is not approximate, this is equal to, so this is nothing but 2 times theta into cos of basically theta square. That is the perfect answer over here. How about this? This is slightly different problem now. This is sine square theta, that means sine theta is squared. Oh, this is just like saying, derivative of sine theta whole square divided by d theta. Why does this remind me of x raised to n formula? Yes, it is. Just that the x is now our sine theta. If it was x square derivative, I would have, if it was x square, derivative, I would have done it properly. Oh, but this is sine theta. So what I will do, I'll use chain rule to now think of sine theta like x. So this will be derivative of sine theta whole raised to 2 with respect to sine theta. And since I divided with something, I will again put derivative of sine theta. And this d theta originally was there. So I put d theta again. I've just used chain rule if you notice carefully. Oh, now it is just simple thing. Guys, derivative of x square with respect to x is nothing but 2x. So hence the answer for this will be 2 times of x. X over here is sine theta. Derivative of sine theta with respect to theta is just nothing but cos theta. But wait a minute, 2 sine theta cos theta is nothing but sine 2 theta. Hence the answer for this is sine 2 theta. That's it. Sine square theta's derivative just came out to be sine 2 theta. What a brilliant question. What a brilliant question. Awesome. And for those of you, Hiroshi and such stupid people out there in the chat box, let me tell you, our DNA batch alone has 1,000+ students. We are more than happy with our students over here. Our other batches also have thousands and thousands of students. So don't even think that Vedantu has shortcoming of students. In fact, there are quite a few centers in Tamil Nadu, Andhra, where we have to stop the admissions because there are no more places in our offline centers. Our Karnataka offline centers are almost full. Yeah, there are only very few batches here and there where few seats are there. That's it. So don't be under that false impression. And also, it happens sometimes that some people do not like Vedantu NEET English to grow because first of all, it's an English channel. So a lot of people have that hatred towards English, especially from certain parts. Number two, some people who have their own vested interest also have this feeling that how come these people are not from this very so-called popular thing or this thing, and these people over here are able to do so well, able to predict the questions in 6 months. When we started the channel, by the way, in December and May exam, how come we were able to predict so many things? How come we were able to complete the syllabus? How come we were able to do revision, everything on the channel? So some people can't digest it. So it happens. I understand it. Maybe you should just go buy a Bernol or something. Or if it is burning somewhere, put some ice over it. Maybe it will help you a little bit. Okay. So just put some ice. Maybe you will feel a little better. Yeah. Okay. Moving on to the next question coming up on your screen. And here it is. Let's find this out. What is the derivative of let's take something crazy, maybe log of 2x minus 3 to the base 5 okay, this is the question which is there in front of you. Okay, so let's find out. Let's find out how much will be the answer. First of all, you cannot find this derivative directly. Reason number one, the base is not e, it is 5. So you need to convert it first of all. First of all, you need to convert it to the natural logs. With that, you cannot do it. So how do I convert it? Remember, chain rule in log, not in derivative, that is separate. Chain rule in log. So what I can do is I can just write this as derivative. Okay, this is to the base 5, right? Okay, I'll do one thing, log of 2x minus 3. Instead of to the base 5, I'll put e. So I'll have to also put log of e to the base 5. This is the chain rule in logarithms. This will be the chain rule in logarithms. Oh, now I don't like e to the base 5, I like to the base e. So I will take the reciprocal of this. By the way, this is just a number. So I can also take that number outside. So it will be log of e to the base 5 into the derivative of, instead of log, I can just write it as ln 2x minus 3. This I can further write it as 1 by log of 5 to the base e, reciprocal rule in logarithms. Here, what I can do, log's derivative is 1 by x, x over here is 2x minus 3, ln x derivative is 1 by x, but this is of the form ax plus b. Whenever it is of the form ax plus b, this number which multiplies with x comes outside automatically. So you have to also multiply it with 2. That number 2 should also come outside. This is the ax plus b rule. This is nothing but ln 5. So this will be 2 divided by ln 5 and this whole thing into 2x minus 3. This is the answer. This is the answer. Everyone? Very good. How many of you got it? Clearo? Perfecto? Are you enjoying this problem solving? Because you can see so many applications of mixed concepts as well. Derivatives, trigonometry, logarithms, everything. Moving on to the next question coming up on your screen. Find the derivative of 5 minus theta whole raised to 8. 5 minus theta whole raised to 8. I'll give you some few things over here as a backup. D theta by dt, rate of change of theta angle with respect to time, is angular speed, which is omega. That is something which you note. Yeah. Okay. So what do you know? What do you do now? First of all, there is theta and there is t over here. I can't take derivative directly. So what do? I'll use chain rule in derivatives. Without that, I can't proceed. How about writing it as, instead of 5 minus theta, I'll write it as minus theta plus 5, one and the same thing, whole raised to 8, derivative of that whole thing with respect to d theta into d theta by dt. I've just used chain rule. Nothing else. You might be wondering, sir, why did you write 5 minus theta as minus theta plus 5? The reason for that is ax plus b rule. See guys, derivative of x raised to n with respect to x is basically nx raised to n minus 1. But when you have derivative of ax plus b whole raised to n with respect to x, okay, then the derivative will be n into ax plus b whole raised to n minus 1, but also multiply it with a. This number a also comes outside. So a into n into this. Basically that coefficient of that x variable term comes outside. Yep. I hope this is clear. I hope this is absolutely clear. So here a is minus 1, b is 5. This is x raised to n format. This is x raised to n format only. So why not write it as minus theta plus 5. 8 will come outside. 8 minus 1 is basically 7. The coefficient of the variable, which is negative 1, will get multiplied. So minus 1. That's it. This is the answer for the first part. D theta by dt, I just told you, is nothing but omega. It's angular speed. So hence the final answer will be minus 8 omega into 5 minus theta whole raised to 7. That is the answer. Minus 8 omega into 5 minus theta whole raised to 7. Perfect. What an answer. What an answer, right? All right. Moving on to the next question coming up on your screen. If it is given to you, that in a step somewhere you might have to integrate something, and the integration is as follows. Find the integration of root of x from -1 to +1. Sorry, -1 most likely will happen. From let's say 1 to let's say 4. Okay, we have to find the integration of this with respect to x. How do we do this? This is a definite integral. A definite integral always gives us the area under the graph. So if somebody draws the graph of root of x, this is x, on the y-axis you draw root of x. That root of x graph might probably look like this. It will be a curved line. So 1 to 4 means this is point 1, this is probably point number 4. I want to find what is the area under this graph. That is what the definite integral actually tells me. Okay, so first do your normal integration. What is root of x's integration? So it is x raised to halves integration from 1 to 4. Now x raised to halves integration is x raised to half plus 1, which is basically 3 by 2, divided by 3 by 2. Limits I will put over here 1 and 4. Now first put 4 over here. 3 by 2 is a constant, bring it outside, so it will just become 2 by 3. Okay, first put 4 over here, so 4 raised to 3 by 2. Then put 1 over here, so 1 raised to 3 by 2, and then subtract it. That's all. So this will become 2 by 3, and then 4 raised to 3 by 2. First take the root. 4's root is 2. So actually this is becoming 2 cube. 1 raised to any number does not matter. It is always 1. What is 2 cube? It is basically 8. 8 minus 1 is 7. So it will be 2 by 3 multiplied by 7, which is nothing but 14 divided by 3. That is the answer. 14 divided by 3. Is that clear? Everyone with me on this? Everybody with me on this? Is that absolutely clear, my dear students? Awesome. Okay, so this is the kind of integration questions that you can get. Let's do some more integration based questions as well. Can we all do the integration of, let's say 1 by 4x minus 1's integration from 1 to 3, okay, dx. Okay, what will be the integration of this? You might be wondering, sir, should I take it on the top and integrate? I would say no. 4x minus 1 is of the form ax plus b. If you remember correctly, integration of 1 by x dx is ln x. So what will be the integration of 1 by ax plus b dx? That would be again log of ax plus b, but instead of multiplying with a, it will now be divided by a. So that's the difference. In derivatives, you multiply with the coefficient of x. In integration, you divide it with the coefficient of that number x. That's all. As simple as that. So what do we do now? Over here, I can see clearly this is a, this is x, this is b. B is minus 1. This is ax plus b form in the denominator. So the answer will be a logarithm. So I can just say it is ln ax plus b, which is same thing, 4x minus 1. The whole thing divided by the number a, which is 4 over here. And the limits of the integration should stick intact, 1 and 3. That 4 is a constant, so just bring it outside, doesn't matter. First put 3, then put 1. So if I put 3 first, so it will be ln 4 into 3 minus 1. This is the first term. Then put 1 over here. So it will become 4 into 1 minus 1. Okay. So that is the second term. And then subtract both of them. So this will become 1 by 4. 4 3s are 12. 12 minus 1 is 11. So ln 11 minus 4 1s are 4. minus 1 is 3. So ln 3 is right over here. So log a minus log b is log a by b. So this will be 1/4 of log of 11 by 3. That's it. That is the answer. You don't have to simplify it further. Okay. I hope this is absolutely clear. Absolutely clear. Everyone with me? Yes. No need of minus sign. Yeah, no need of minus sign, Krishna. Minus sign is not going to come over there. Yeah. Unless you wrote something. Acha, you wrote log 11 minus log 3. Okay, okay. Log 11 minus log 3. Okay, okay. So minus, I can combine it as division in logarithm. That is one simple rule which you can do. Okay. As simple as that. All right. Next, let's go to the next question, which is going to come up on your screen. Let's see if we can do the integration of. Let's see if we can do the indefinite integration of. Let's take something like 5 times e raised to sine theta multiplied by cos theta d theta. Can you do the integration of this? Okay. Very interesting question, guys. What will be the indefinite integration of this? Okay. It looks like a very scary problem at the start, but actually it is not. Actually, it is not. We'll see why. You can think of some substitution which will simplify it. Whenever you see somebody's derivative is present inside the integral itself. So over here, sine theta's derivative is cos theta. And when you see such things happening, what you can do is you can substitute that variable or that part of the integral whose derivative is present. Sine theta's derivative is present. So why not substitute sine theta only as x? Therefore, derivative of sine theta with respect to theta will be nothing but dx by d theta, both sides differentiating with respect to theta. Derivative of sine theta is nothing but cos theta. So cos theta is basically dx by d theta. Oh, interesting thing is cos theta d theta is nothing but dx. I can see cos theta here, I can see d theta here. Cos theta d theta is present, so I just can put it as dx. What about the rest of the things? 5 is just a constant, put it outside. Integration of e raised to sine theta. What is sine theta? Sine theta is also x. Oh, e raised to x is integration. Everybody knows it is the same thing. So it will be again e raised to x plus c. But towards the end, never leave the answer in terms of x because it was an assumed variable. Leave it in the original variables, which was theta. So x was nothing but sine theta. So 5 times e raised to sine theta plus some constant. That is the final answer. Yes, substitution method. Got it? How to do this problem? Very good. Excellent, my dear students. All these are amazing, amazing questions. How about these questions? Check this out. P hat plus Q hat plus R hat is equal to 0. Question is, find the angles between all the vectors. All the vectors. That is the question. Okay, find the angles between all the vectors, P hat, Q hat and R hat. So what is this P hat? Unit vector, Q hat unit vector, R hat unit vector. But the problem is, you know, I do not know how they are aligned. But one thing I know, that their sum, vector sum is 0. So how about thinking about one vector here, another vector here, another vector here. Basically, it should complete a closed polygon like this. So only for a closed polygon, only for a closed polygon, only for a closed polygon, the vector sum is 0. The vector sum is basically 0. So it should form a closed polygon, which is a closed triangle. But the best part, P hat, Q hat and R hat are unit vectors. That means the side length of that triangle is 1. Each side is 1. So if all the sides are 1, it should be an equilateral triangle. Oh, So sir, this is like this, this is like this, and this is like this. So this is P hat, this is Q hat, this is R hat. So this is nothing but an equilateral triangle of the side length 1. Now all the internal angles are 60 degree. We all know that, but that is not the correct answer. Be careful about it. Between P hat and Q hat, you can't measure the internal angle because the head and tail are coinciding. So shift them properly, slide the vector P hat, slide the vector P hat, and let that Q hat be over here. So this is the correct angle, which is 120 degrees. Same thing between R hat and P hat. So what you can do, slide that R hat downwards like this. This is P hat. Slide that R hat over here. So this is the correct angle, which is 120 degrees. Between Q and R, again slide that Q over there. Slide that Q over there. R hat is as it is. Slide that Q over there. Okay, slide that Q over there. This is Q hat. If that internal angle was 60, this will be again nothing but 120 degrees. So the correct answer for this question is 120, 120, 120, and 60, 60, 60 will be the wrong answer. 60, 60, 60 will be the wrong answer. Everyone got it? Very good. Awesomeness. Keep it up. Keep it up. Moving on to the next question. If I tell you, find the angle between I cap minus j cap plus k cap and 2 I cap plus 2j cap minus 2k cap. What is your answer going to be? Okay, find the angle between I cap minus j cap plus k cap and 2 I cap plus 2j cap minus 2k cap. So you have to find the angle between these two vectors, and whenever you have to find the angle between two vectors, think of dot product. Think of dot product. So these kind of questions are also again common. So cos theta is a vector dot b vector upon the magnitude of vector A and magnitude of vector B. That's how it goes. So when you take the dot product of these two vectors, I cap minus j cap plus k cap, dot product with 2 I cap plus 2j cap minus 2k cap, whole thing divided by magnitude of the first vector will be root of 1 square plus 1 square plus 1 square, multiplied by magnitude of this vector will be 2 square plus 2 square plus 2 square. What is the dot product of this? I with I, 1. So 2 will be the first answer. Minus 1 into plus 2 is minus 2. Plus 1 into minus 2 is again minus 2 over here. The whole thing divided by, what is this? This is just root 3. What is the next one? It is nothing but 2 is coming outside. 2 is coming outside, and root of again 3 over here. 2 minus 2, 0. So hence it will be minus 2 divided by root 3 into 3. Root 3 into root 3 is 3. Multiplied by 2, 2 cancels. So hence the answer will be negative 1 by 3, but that is cos theta. So therefore, theta will be negative cos inverse of negative 1 by 3. That is the answer. Cos inverse of negative 1 by 3. Is that absolutely clear, my dear students? Everyone with me? Cool. Got it? This is how you find the angle between any two vectors. Awesome. 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 Let's do one more question. Let's do one more question coming up on your screen. Here it is. Here it is. Let's start doing one more question, guys. If I tell you to find the value of A cross B dot A hat, what is the answer going to be? What is the answer going to be? A cross B dot A hat. Come on, think about it. First of all, think like this. If I have a vector A and some another vector B in some plane, and I find out what is A cross B, A cross B will be a vector like this using right hand rule. This will be A cross B. The best part is it will be perpendicular to B also and A also. So 100% A cross B vector is simply put perpendicular to A and B both. A and B both using right hand thumb rule. So whenever you have two vectors which are perpendicular, and if you take the dot product, what happens? It's perpendicular to A cross B. So if you take the dot product of them, what do you think will happen, guys? A cross B dot A cross B. If you take the dot product, A cross B dot product, if you take it with A, what do you think the answer will be? It will be a big fat 0, right? Because perpendicular vectors cos 90 is 0. Remember that. So cos of 90 degrees is just 0. Hence the answer is also 0. Very good. Very good. Is that absolutely clear? Very nice. Very nice. Keep it up. Let's see whether you can get the next few questions also. Which of the following are mathematically, correct or wrong? Some of them are mathematically only wrong. Some of them mathematically they are fine. Okay. So which of them are correct? Which of them are wrong? Options are something like this: A bar plus B bar minus 2 times C bar, A bar dot B bar dot C bar, A bar plus B bar dot C bar minus D bar, A bar dot B bar plus B bar dot C bar, okay, or B bar dot D bar, okay. Then A cross B cross C, A cross B dot C, A dot B cross C, okay. Lastly, A dot B plus C cross D. Okay. Some of them are mathematically correct, some of them are mathematically wrong. Which of them are they? Okay, just think about it. Just think about it. Let's do the math simply. See, guys, I have a vector added with another vector, added or subtracted with another vector. That's perfectly fine. There is nothing wrong in this. So this is absolutely correct. Nothing wrong over there. This over here, on the other hand, I feel is a problem. I'll tell you why. A dot B is actually a scalar. A dot B is actually a scalar, and you cannot take a dot product with a vector. How is that possible? Whenever there is a dot product, on both sides you should have vectors. Not one scalar or two scalars. Both sides should be vectors. Hence, this is mathematically only wrong. Mathematically only wrong. Nothing correct over there. Here, A plus B is a vector, is nothing but a vector, and C minus D is also a vector, and you are taking a dot product of both of them, vector dot vector. There is nothing wrong in it, so this is perfectly okay. This is perfectly okay. What about this? What about this? A dot B is nothing but a scalar, like number 2, number minus 3, something. This is also a scalar, and I'm just adding both these scalars. There's nothing wrong in adding scalars. This is perfectly okay. I can add two scalar numbers, like 5 plus 7 is 12. Nothing wrong in it. This one, what is it? This is nothing but a vector. A cross B is nothing but a vector, and I'm crossing it with another vector. I'm crossing it with another vector. A cross B is just a vector. Here, the C cross D is just another vector. Where are the scalars over here? A dot B is just a scalar. A dot B is just a scalar. Okay, now vector cross vector, no problem. It is perfectly okay. You can definitely do it. Here, vector dot vector, definitely I can do it. C is also a vector. C is also a vector, and there is a dot product in between them. There is nothing wrong in this. This is also perfectly okay. This is a problem. How can I do scalar cross vector? How can I do scalar cross vector, guys. This is wrong. It is not allowed. Scalar cross vector. Even this is not allowed. How can I add a scalar with a vector quantity? This is also wrong. This is also going to be wrong. See if this is clear. Okay, so that was the answer for this. Now I think it's time for chemistry. I think Diksha ma'am will come in. So let me just ask Diksha ma'am to join in.
Hi students, welcome to Vedantu NEET English. I am 𝐂𝐡𝐞𝐤 𝐎𝐿, your chemistry master teacher. And as you have completed bio part, you have completed physics. But now we are going to start with the chemistry. So everybody, am I visible to all of you? Bacha, am I visible? Everybody in the chat box, welcome chemistry with a very beautiful smile on your face. Yes, bacho? Everyone, yes, what we are going to discuss, we are going to not discuss anything here. We are going to revise things with the help of simple basic questions, right? That are there in your NEET exam, right? So these are basic questions that are very simple, but we have covered. So I have picked most of the questions from mole concept, from periodic classification, and from structure of atom that you have completed with me only, right? Okay, bacho? So this way you will be able to identify all the things here if you are able to, recall things back from that, and the questions are simple. The questions are easy, yet the questions will, these questions are only those kind of questions that were there in your NEET exam. Okay, everybody, green hearts in the chat box for your own Diksha ma'am, and let us start the session. Are you all ready? If you are ready, like this, like with this josh, with that punch. Okay, so I want all of you to give me green hearts in the chat box. Yes? Okay. Chalo, let us start the session without wasting any time, and also don't forget to like this session. Yes, don't forget to like the session. Okay, and also we wait for your comments. Yeah? Why don't you guys comment? What is this laziness, huh? What is this laziness? So if some patient asked you to give the prescription, what you will say? I'm so lazy. I don't used to comment on my teacher's video. How can I give you these medicines and all? I won't write anything. You only, I will just speak something. You can just identify those things. No. You have to write, no. Only then we will be able to understand what you want. Okay? Chalo. Let us start the session. The very first question First question is Rutherford experiment on the scattering of alpha particles. Right? Now Rutherford did an experiment for alpha particles. Right, bachcho? So what was that alpha particle experiment? Firstly, what is that alpha particles? Alpha particles are beryllium-2 positive, are helium-2 positive, right? Which basically means it has 2 protons and 2 neutrons, right? That means if you take out 2 electrons from helium atom, that becomes a alpha particle, and these alpha particles are positively charged, right? So Rutherford basically constructed this experiment, and what he concluded out of that there is a nucleus. Yes, very good. And now tell me everybody, nucleus, were neutrons also discovered by Rutherford or not? Were neutrons also discovered by Rutherford? No, 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 no. Neutrons, he just said that, okay, there is a very small positive thing inside the nucleus. For example, there is big stadium, and inside that there is just Virat Kohli, little Virat Kohli, chotu motu, right? He is the nucleus, right? So that nucleus is positive in charge and it has proton. He had no idea about neutrons. So neutrons were discovered by Chadwick, not Rutherford, okay? So in this model only, yes, C for capto, that is nucleus is the right answer. Next question is, which of the following is not the fundamental scientific quantity? Which of the following is not the fundamental scientific quantity? Guess, bacha, which was not the fundamental quantity? Volume, mass, time, length. So basically we have 3 fundamental quantities, right? First is length. That is in meters. The SI units are meter. Then we have mass. The SI units here are, the mass, SI units are kilogram, right? Then we have time. This is in seconds. Rest of the quantities are derived from these 3 quantities. For example, if I talk about volume, that is length into breadth into height, right? If I talk in maths, that is length into breadth into height, right? Or if you talk about sphere or anything, that will be multiplication of something, right? So basically, even if we talk about density is equals to mass by volume, even then it's not pure fundamental quantity, right? So basically, these 3 are fundamental quantities. Then that means volume is not a fundamental quantity. Answer is going to be 1. 1 is going to be the right answer. Yes, bachcho, very good, shabash. Next question. Which of the following pairs of compounds illustrate the law of multiple proportions, right? Which illustrate the law of multiple proportion. Now, ma'am, what is this law of multiple proportion? Firstly, you guys answer, then I will answer. Okay, firstly, though, I want to see your answers in the comment section. Whether it's H2O and Na2O, whether it's MgO and Na2O, whether it's Na2O, BaO, or SiCl, SnCl2 and SnCl4. So you have 4 options. Yes, very good. The option you are saying is 4th option, right? You are saying the 4th option. Yeah? Just wait a minute. Yes. So you are saying the 4th option. So, bacha, what is this law of multiple proportion? Law of multiple proportion. Multiple proportion. If I talk about this law, this simply means that multiple proportion means you have 2 elements, for example, carbon, oxygen. They can form in different proportion. Multiple means more than 1 proportion. If it combines with 1 carbon and 2 oxygen, it can form a different compound, that is carbon dioxide. If it combines with 1 carbon, 1 oxygen, it will form carbon monoxide. These 2 compounds are completely different from each other. For example, I have hydrogen, oxygen. So when they combine together, hydrogen, if 2 hydrogen and 1 oxygen, it will form water. If 2 hydrogen, 2 oxygen, it will form peroxide. So that is a completely different molecule as compared to H2, right? So similarly, that means here we need to identify which has same number of, same atoms, right? So this is tin, this is also tin, this is chloride, this is also chloride. So these 2 are the these two belong to the law of multiple proportion, right? These two belong to the law of multiple proportion, right? Okay, yes, very good. Next is, out of these two, are isoelectronic, isotones, isobars, or isotopes of each other. So now these iso species are very important, right? These iso species are which are very important. Now firstly, let me tell you the different definitions of these species. So basically these four species, I have also told you, isosters, isodiaphers in the structure of atom. Those are also very important, right? Then what is isoelectronic? Where number of electrons, iso means same. Here number of electrons are same. Isotones means number of neutrons are same. Number of neutrons are same. Isotopes means you have atomic number is same but mass number is different. But mass number is different. Okay, mass number will be different here. Here when we talk about isobars, that means atomic number is different. Atomic number is different, mass number is same. Mass number is same. Okay, so if we talk about these two species, that is this and this. Now isoelectronic, this is not possible. It has 6 electrons, it has 8 electrons, right? So that is so not possible. Isoelectronic, not possible. Isotones, for that we need to calculate the neutrons here. That is A minus Z. Okay, if I represent my element by this is X, A, Z. So A is basically neutrons plus protons. What is this Z? That is nothing but protons, right? Or in a neutral atom, this is also equals to electrons. So that means to calculate neutrons, that will be A minus Z. A minus Z is 14 minus this, it has 8 neutrons. It also has 8 neutrons, right? That means they are isotones of each other. B for Baswa sir is the correct answer. What about isobars? Right, example I can tell you, example is like carbon, examples I will tell you, right? For example, carbon 614 and nitrogen 714. Now they have same mass number but number of protons or atomic number is different, right? Here when we talk about this, it can be carbon 612 or you can say carbon 613, carbon 614, or these are isotopes of each other. Okay? Yes, Pacho? Next question is very important question, right? Now, which of the following is the largest in size, right? They are asking about the size here, right? They are asking about the size here. So this we have done in periodic classification and this is from the base of chemistry. You can understand this with the help of a concept, right? So which of the following is the largest in size? Firstly, what we will see in these cases, whenever some ions are given to you, check whether they are isoelectronic to each other or not. So very first thing that you need to do is check whether they are isoelectronic or they are not isoelectronic. So how many electrons here? It has 17 plus 1, that is 18 electrons. How many electrons are here, bacha? It has 16, sulfur is 16 plus 2, that is 18 electron. That means these two are isoelectronic to each other. Let us talk about sodium. It has 11 atomic number, plus means it will be minus 1. It is a cation that will be formed by losing 1 electron. So minus 1, that is 10 electrons, right? That is 10 electrons. What is this fluorine? Fluorine belongs to 9 atomic number, right? Beta 1 to 30 atomic number should be on your tips, okay? Should be on your tips. So today in my Telegram channel, 𝐂𝐴𝐤 𝐎𝐿 Chemistry, I will send you 1 to 30 atomic number along with the mnemonic of learning that, okay? 1 to 30 atomic number, right? 1 to 30 atomic number, the element and the way of learning the atomic number and their masses also, okay? So I will tell you that, okay? I will send you a page there. Now, this is 9 plus 1, that is 10. So, these two are isoelectronic to each other. Now, bacha, always remember, more are the electrons, more is the size, right? Because here you have 18, here you have 10. Just there is a jump of 1 shell here, right? There is a jump of 1 period here. That means what we will say, definitely these two are not going to be the largest in size. The competition will be between Cl negative and S2 negative, right? So firstly, I will tell you a logic behind that, then I will tell you a simple trick behind that, right? So firstly, though, you can imagine that in Cl negative, right? In Cl negative, beta, everybody in the chat box, how many electrons you have? Cl negative, this is S2 negative. How many electrons you have? 18 electrons. It also has 18 electrons. How many protons, beta? Protons are atomic number only. This has 17 protons. This has 16 protons, right? And we want to see the Z effective. That means for less, it's like two villains, one hero and one villain is fighting with each other, right? So this has less proton are attracting the same number of electrons. It has one more proton is attracting the same number of electrons. So here Z effective. Now here it will be able to attract these 18 electron with more efficiency, efficiency. Okay, I don't know, so sorry. Yeah, so here beta it will be 16 proton. 16 proton will be attracting, that will not be that much powered here. Why? Because you can imagine that here you have 16, here you have 17, here you have 16. Number of electrons are same. Here Z effective is more, so it will be able to pull the electron with more force. It won't be able to pull the electron with that much force as compared to Cl negative. So size is going to decrease here. So size will be like this. So size will be like this. So the answer will be 2 is the correct answer, right? 2 is the correct answer. Okay. Now second important thing here is, ma'am, we will get confused here. Ma'am, please give us some easy method of doing this. So I have easy. First is concept. What is my way of teaching you? Firstly, we will learn this with the help of concept. Then we will learn this with the help of trick, so that you can do that easily in your exam hall, right? So here, bacha, what you will do in isoelectronic species, only in isoelectronic, beta, only in isoelectronic, right? You will simply write that it will be equals to atomic size will be inversely proportional to atomic number. It will be inversely proportional to atomic number. As atomic number of sulfur is less, so here its size will be more, right? This trick is based on this particular concept only that I have already taught you just now. Okay, bacha? Easy, and this question is very, very, very important. You can directly see these kind of questions in your exam hall, in a NEET exam. Okay? Next question, beta. Which of the following is statement is connecting the 4 quantum number is false? So beta, everybody in the chat box, be active here. Let's draw that simple quantum number table. Who have not gone through the lecture of my quantum numbers, beta, please go through that. I have done that in detail. I will also take up one more lecture based on that, where we will talk about different quantum numbers, right? Okay. Okay. Why it is happening like that? Okay. Now, okay. So I'll give you just few things about it, right? So N, L, Ml and S, right? These are the 4 quantum numbers. Principal quantum number, that defines the shell here, okay? This is a very quick revision of that. This defines the shell, this defines the, this is, I don't know why this is happening, right? Wait. I don't know why this thing is so irritating, right? These ads are so irritating. Okay. Now, beta, let us talk about these 4 things here. Okay. When we talk about N, that is shell, L is subshell, right? Ml is orbital, then S is the spin, right? If we talk about shell, that tells us about the size and the energy. L, that means subshell. That tells us about the shape. That is whether it's S, whether it's P. Okay. because it is going to be 0, 1, 2, so on up to n minus 1. Okay, this is going to be 1, 2, 3 and so on up to infinity, right? So it is going to tell us about SPDF, that is S, P, D and so on F, right? Okay, 4 will be your, 3 will be your F, right? So it is going to tell us about the shape of the orbital where it is, right? Then what about this ML? What this ML is going to tell us about? When we talk about this PX, P, we talk about PX, we talk about PY, PZ. In D, we talk about 5 orbitals, dx square minus y square, dz square. So these are x, y, z. These are what? These are the coordinates. So this orbital is going to tell us about the orientation. Okay, this is going to tell us about the orientation, right? And this ML will be equals to minus L2 plus L, right? In the detail, I have already taken up the lecture for this. Then, bacha, what about this spin? Spin is nothing but it is going to tell us about the clockwise or anti-clockwise direction, right? Okay, this will be minus half or plus half, right? Okay, now based on these things, you tell me which is the correct answer. Based on these things, N gives idea of size of the orbital. Yes, N gives us the energy and size, right? L gives the shape of the orbital. Yes, that is also true. It gives us the shape of the orbital, whether it's spherical shape, it's dumbbell shape, it's double dumbbell shape, right? Then what about M? M gives the energy, whether it's true or false. ML, that gives us the orientation, not the energy. Energy is given by N. More is the value of N, more is the energy, right? So answer, this is not correct, right? This is not correct. This gives us the orientation, right? This gives us the orientation. S gives the direction of spin of electron in an orbital. Direction of spin clockwise or anti-clockwise, that is also true. They were talking us about the false statement here. So that is going to be the third statement, that is the false statement, right? Okay. Yes, bacho. Next question. Number of electrons present in 10 gram of DO2 is. Now let us see this. Let us see this question. 10 gram of DO2. Bacho, 10 gram of DO2. Everybody in the chat box. 10 gram of DO2. Everybody, 10 gram of DO2, what does that mean? Number of electrons. What is this? Beta, this DO2 is the heavy water, right? Remember this hydrogen. Hydrogen has 1 proton and 1 electron. Deuterium has 1 proton, 1 electron and 1 neutron. Okay, hydrogen does not have any neutron. Deuterium has 1 neutron, right? So remember this thing, right? Electron is same, neutron is different here, right? So mass is different, right? That means mass is going to be different. Here the mass is 1, here the mass is 2. Here the electrons is 1, here also the electron is 1, right? So firstly, let us calculate whenever they ask you anything about number of molecules, the volume, anything. In mole concept, you always calculate the number of moles, right? So firstly, let us calculate number of moles here. That is going to be given mass, that is 10 divided by molar mass. Molar mass here is D, that is deuterium, has mass 2, right? So it will be 2 into 2 because it has 2 atoms plus 16, right? That is the mass of oxygen. That is 10 plus 4 plus 16, that is 10 by 20, that is beta 1 by 2. Okay, here number of moles is 1 by 2. Now then calculate the number of, you can calculate the number of electrons. How will you calculate the number of electrons? Firstly, calculate number of molecules. Okay, number of molecules. Number of molecules is number of moles multiplied by Avogadro's number. Okay, number of moles multiplied by Avogadro's number. That is the number of moles, right? That is the number of moles. Then after calculating the number of moles, you need to calculate the number of electrons they are asking you. So you can say one molecule of D2O, how many electrons? 2 electrons from, see, you can say 1 electrons from one deuterium, 1 electrons from other deuterium, 8 from this oxygen. So it will be 8, 9, 10 electrons, right? So you will say one molecule has 10 electrons, 1 by 2 Na of D2O will have 1 by 2, these many molecules now you have, into 10 electrons. That is 5 Na of electrons, right? That means how many electrons you will have? 5 into Avogadro's number of electrons. That is C, that is third is the correct answer. Okay, that is, this is the correct answer. Whenever they ask you anything about this, just calculate the number of moles. After calculating number of moles, calculate the number of molecules. That will be number of mole multiplied by Avogadro's number, that is 0.5 into Na. Then you know in one molecule you have 10 electrons, right? So in these many molecules, how many electrons? It's like you have in one chocolate you have 2 pieces of nuts. Then in 10 chocolates, how many pieces of nuts? That will be 10 into 2, that is 20. Same thing is here. Okay, same thing is applied here, that is C is the correct answer. Now this question beta, helium and beryllium have, everyone, what is the answer for this question? Everyone, what is the answer for this question, everybody? Okay. Helium and beryllium have similar outer electronic configuration. This is what they are saying. Okay, ma'am, let us see. Helium, helium, helium. This is our helium. Helium is atomic number 4, right? Atomic number 2, right? So it is 1s2, right? 1s2. What is beryllium? That is atomic number 4, that is 1s2, 2s2. Okay, yes. So both of them have electronic configuration ns2 type, that is, so we can say this is so true. Okay, we can write this is so true. Both are chemically inert. Helium is a noble gas, but beryllium, this is not a noble gas. This is a noble gas, right? So both are not chemically inert. So answer is, assertion is true, but reason is false, right? So answer will be 3 is the correct answer, right? 3 is the correct answer. Any problem here, bachcho? Any sort of problem that is clear to everybody? Yes, bacha, if any doubt you feel anywhere, you can always ask me without any hesitation. Okay? Yes. Next question, bacha, we have number of protons. This is also a very, this is also asked so many times, right? This is your PYQ only, right? The number of protons, neutrons and electrons is this respectively, right? So, bacha, let us see. It has, for example, any element, that is X, A and this is Z. A is, as I told you, that is the mass number, that is number of neutrons plus number of protons. What is Z? That is proton, that is atomic number, and for a neutral atom, this is a neutral atom, this Z proton will be equals to electron. This is only for a neutral atom, right? Otherwise it will not be. Now, can I say that number of neutrons will be A minus Z? If I say A minus Z, that is neutrons plus protons minus protons, that will be neutrons. So can I say neutrons will be A minus Z, right? So what is A? What is A? That is 175 minus 71, that will come out to be 104. Okay, that will come out to be 104. What is number of protons? Protons will be this Z only, that is 71. What is electron? That will be this Z only, that is 71. So answer will be protons is 71, right? Neutrons is 104. Proton is 71, neutrons 104, electrons is 71. Third is the correct answer. Okay. You can also do this in 1 second. How you will do it in 1 second? Just see protons and electrons. Protons and electrons, that is should be 71, and this is the only option where protons and electron is 71. You don't even need to calculate neutrons here, right? Okay. but still I taught you, I taught you because, you know, it's like not the same question will be repeated in your exam, right? So maybe in the option you might need to calculate neutrons, and this, so you should know how to calculate neutrons, right? It's a very, very simple yet very important question. Now, bacha, this question, 1 mole is the amount of substance, 1 mole is the amount of substance that contains as many particles. Basically, they are asking about the definition. This I expect from you that you will give me the answer within 1 second. Yes, bacha, that too I definitely expect from you. Yes, they are asking the definition. 1 mole is the amount of substance that contain as many particles or entities as there are atoms exactly in. Firstly, so beta, there is no carbon 13, no carbon 14 isotope, right? So this will be to eliminated, right? This I told you in 1 mole, right? So 1 mole is the number of, that is Avogadro's number, that is present in 12 gram of carbon 12 isotope, right? So answer will be this, okay? Answer will be 12 gram of carbon 12 isotope. The number of elements, that is nothing but, number of atoms, that is nothing but Avogadro's number, okay? Yes? 1, 1, 1, 1, 1, yes. Which of the following electronic configuration is correct explanation of Aufbau principle? Which of the following is the correct? Okay, everybody, interesting question. Correct explanation of Aufbau principle. Do you remember what is this Aufbau principle? So while you are solving, I will tell you what is Aufbau principle. Aufbau principle, beta, that is N plus L rule. More is the value of N plus L, more will be the energy. If N plus L, this is for multi-electron system, okay? If N plus L is same, then more is the value of N, more will be the value of energy. From there, we also calculated the energy level diagram, right? We also did the energy level diagram. So, by that, what can you say about this, right? 1s to 3s2, no, this is not energy level diagram. 1s to 2p, definitely not. 1s to 3s2, no. It is 1s to 2s to 2p, then after that 3s, then 3p, right? This is how it goes on, right? Okay? Yes, bacho, done. Done, everybody. Bacha, if you want to calculate the value of energy level diagram, I have a simple trick for you that I have told already. Students, I have already told you. SPDF. What you do here? 2 times S, 2 times P, then 2 times S, then 2 times PS, 2 times S, then 2 times PS, right? Then 2 times DPS, 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 then 2 times FPTPS, right? FDPS, FDPS, right? So now this will start from 1, 2, 3, 4. Starting S from 1, this is 2, this is 3, this is 4, this is 5, this is 6, this is 7, right? Starting P from 2, that is 2, this is 3, this is 4, this is 5, this is 6, right? Starting D from 3, that will be 3, then it is 4, then it is 5, right? Then starting F from 4, that will be, this is 4F. So this is going to be your energy level diagram, right? Okay, this is going to be your energy level diagram for this, right? And without this, inorganic chemistry is totally impossible, okay? That is totally impossible. Now, what about this question, bacho? What about this question? The molality of a solution does not change with change in temperature. Molality we are changing, right? Always remember one thing, all the terms that are dependent on volume will be dependent on the temperature, right? Terms which are independent of the volume will not be dependent on the temperature, right? So molality, as you know, molality, the definition of molality is number of moles of solute or you can say number of moles of solute divided by, that is molality is mass of, you can simply say mass of solvent in kilograms, right, in kilograms. So that is why it is independent of volume, and that is why it will not change with the temperature. This is the true thing. Molality is why? Because molality is expressed in terms of mole per 1000. 1 kilogram means 1000 grams only, right, of the solvent. So this is also true, and this is the correct explanation also. So answer will be 2. Second will be the correct answer. Why third? Because second is the correct answer because of this formula only. In this formula, I cannot see anything related to volume, and that is why this term is independent of volume, and that is why this is independent of temperature. So this is the correct explanation. Okay, so answer will be 2. Okay, answer will be 2 is the correct answer. Okay, yes. Okay, bachcho, 2 is the correct answer. Very good. Next is, which of the following oxides is most basic? Which of the following oxides is most basic? Now you know in a periodic table what exactly happens, right? What exactly happens in a periodic table? You can see that here, from here you have metalloids, right? Here you have non-metals, here you have metals, right? Here you have metals. And you already know that down the group metallic character increases, and along the period metallic character decreases, right? So from here, which will be, so when you're talking about most basic, right, basic character of an oxide is dependent on the metallic character. It is dependent on the metallic character. More is the non-metallic character, more it is acidic in nature, right? So as you know, which has the strongest metallic character, that is somewhat here, that is here, like aluminium is also here, sulfur is somewhat here, silicon is also there, right? So only sodium is present in the most metallic side. That means it will have the most acidic character, most basic character, right? So answer will be 1 is the correct answer. Okay, 1 is the correct answer. Very good, beta, very good. Now attempt this for classifying elements by plotting the atomic masses of elements against the volume was made by. This means they are saying there was a graph that was between which thing? That was between atomic masses and elements, of plotting the atomic masses of the element against the volumes, right? Against the volume. This is the mass, atomic mass, this was the volume. So the graph, I will give you a hint about it. The graph was something like this. Okay, it was something like this. The graph was something like this. We also felt sad about him, right? Who was that guy? Mendeleev also gave the same thing, but that was in the form of table. He gave the same conclusion in the form of a graph. What was that? That person was Lothar Meyer. Okay, the person was Lothar Meyer. He gave this graph, right? From this chapter, there also can be one more graphical question. That can be, there was a graph made between under root of frequency and atomic number. This graph was given by whom? Okay, this graph was given by Lothar Meyer. Lothar Meyer, right? What about this graph? This graph was given by whom? It's not in the options. This graph was given by, this particular graph was given by Henry Moseley. Henry Moseley. That is a straight line graph he gave, right? He gave a straight line graph. Okay. On the basis of this graph only, there is the formation of this modern periodic table. So someone asks you from which is the base of modern periodic table, that is Henry Moseley graph. Okay. Yes, bachcho, there are only 2 graphical questions that can be there, asking you whether this graph was made by whom, right? Only these 2 graphs. Rest are the trends are important. Okay. Yes, bachcho. Next question. Which of the following is considered as metalloids, right? Which of the following is considered as metalloids? Okay, you have to, like, they are between in P block, in P block, here you have these elements, right? Here you have these elements, and these elements are all of them, right? These are metalloids, okay? These two are metalloids, these are also metalloids, these are also metalloids, okay? So all of these, you have to remember this, all these are metalloids, and like along the period, away from metalloids you have non-metals, and before beta, after the metalloids you have non-metals, before the metalloids you have metals, right? Okay, so all of these is the correct answer here. Yes, D for doctor, D for Diksha ma'am, D for dosa is the correct answer. The fifth period of modern periodic table. This is also a very basic question, right? Very basic and very important question. Very important question. Fifth period of modern periodic table includes how many elements? Includes how many elements? 18, 5, 32, 21. I know you won't be confused between 5 and 21. This so definitely not. Right? Students will do mistakes in 18 and 32. Now you tell me, out of these two, which is the correct answer? Right? Yes, these are our rock stars. Right, Gopika ma'am? Right, Gopika ma'am? These are rock stars. They can sit for hours and hours and study. This is the quality of a future doctor. This is the quality that a future doctor can only hold. That is this much concentration, right, with full physics, chemistry, bio continuously. Yes, 18 elements. Now students, see this. If you talk about first period, second, third, fourth, fifth, sixth, seventh. Now see, in the first period you have 2 elements, right? In the second period you have 2 plus 2, that is 2 plus 6, 6, 7, 8 elements. In the third period, right? In the third period you have again how many elements? 6 plus this, that is 8 elements. Right, in the fourth and the fifth period, you have 10 elements of D blocks will also be included here. Right, here F block will also be included, so that will be 32. It will also be 32. See this, in the first there will be hydrogen and helium only. Right, in the second there will be S and P. S block will have 2 elements, P block will have 6 elements. Here also S plus P, right? Here it will be S plus P plus D. Right, here D block will start. D block is like this, na? It's like this. So from here, from the fourth period, D block is going to start. Okay, so it will also be S plus P plus D, and here you will also include 14 elements of which 14 elements of D F block, inner transition elements. Right, that is after the lanthanoid, after the lanthanum and actinoid. S, P, D and F. Here also it is S, P, D and F. This can also be done on the basis of magic numbers, right? Okay. Yes, so these are the elements. This is how you need to remember all the periods, how many elements are there. Okay. So that is, here you have, in the fifth period you have 18 elements. That is, A is the correct answer. First is the correct answer. Yes, very good. Next one, bacha, you have, which of the following can act as both Bronsted acid and Bronsted base? Which of the following can act as both Bronsted acid and Bronsted base? The concept of acid and bases. The concept of acid and bases. What is this concept? See, Bronsted acid means, Bronsted acid beta, Bronsted acid, you need to know all the concepts here. Bronsted acid means which will give H positive, right? It is going to release H positive. Bronsted base, that is going to accept H positive. It is going to accept H positive. So beta, what is this going to be? It is going to give H positive. Now, can it give H positive? So it cannot be in Bronsted acid. Can this give H positive? It cannot donate H positive. Donate H positive, not possible. It can also donate H positive, not possible. right? This is also not Bronsted acid. It can accept, it can also accept. They cannot give H positive, so they are not Bronsted acid. They are asking both Bronsted acid as well as base. Now let us look at this one. If it loses, right, if it loses, you have this thing, you have CHO3 negative. If it loses H positive, that will become CO3 2 negative, right? If it accepts H positive, right, if it accepts H positive, it will become H2CO3, right? You can see this, na? H positive means negative and positive will neutralize, right? So it will be CH2O3. Nothing, you can write it as H2CO3, right? This is the mineral acid. Both of them are possible. H2CO3, this is a mineral acid and this is a carbonate ion. Both of them are possible. So that means this can act both as acid and base, right? Now, ma'am, what about this H3O positive? What about this? So many of you are answering this as the correct answer. So now let us see this. This is H3O positive. Now if it loses H positive, it will be H2O. This means it can act as a Bronsted acid. It can lose H positive. If it accepts H positive, it will be H4O2 positive. Have you ever seen something like this? Have you ever seen something like this in your life? I have never seen. If you have seen, then please tell me. Have you ever seen something like this in your life? H4O2 positive. Is it possible? This does not exist. There is nothing, no ion like this is possible, right? So that is not possible. That means it cannot act as, it cannot accept H positive, not possible. So this is not a Bronsted base. Only this can be a Bronsted acid as well as Bronsted base, right? So second is the correct answer. Understood, bachcho? So 2 is the correct answer, right? Okay. Next is, which of the following species is an acid and also conjugate? Ma'am, we just talked about Bronsted. Now what is this conjugate? Conjugate, beta, that means, for example, I have HCl, right? Now this is an acid, right? When it loses H positive, it will become Cl negative. Now this is a conjugate base of this acid. This is a conjugate base, okay? This is a conjugate base of this acid. Okay, understood? So this is what we are going to see here in all these cases. So first is HSO4 negative. They are saying, is this a conjugate base of something? Yes, if we talk about H2SO4, when it loses H positive, it will become HSO4 negative. When we talk about H2SO4, if it loses H positive, this is the conjugate base of this acid, right? This is the conjugate base of this acid, right? So this is what they are saying, which of the following species is an acid, and it itself is an acid. It also loses H positive. It also loses H positive and becomes SO4 negative, right? That means this species is an acid to this one, it is an acid to this species, and it is a conjugate base to this species. That means it can act as, it is both an acid, it is an acid and also conjugate base of another acid. So right answer will be this. Is this possible for any other option? Let us talk about second. Is it possible that there must be some ion like H3SO4 positive? After losing H positive, we got H2SO4. This does not exist only, right? So this is not a conjugate base of anything, right? So this is not possible. This is not a conjugate base of anything available. This does not exist, not possible, right? Let us talk about the third option, OH. Yes, this is formed by H2O. It loses H positive and then we have OH negative. That means it is a conjugate base of water. But is it itself an acid? OH negative, it's a base. It's not an acid. It's hydroxide, right? So that is not possible. Again, this is cross, right? Again, this is cross. What about H3O? What about H3O? What about the fourth option, that is H3O positive? What about this option? Can it lose H positive? For example, we have H4O positive. After that only, after losing 2 positive, after that only losing H positive, it can become H3O this, right? Only then it can become H3O positive. That means if this is a conjugate base, first thing that this is not even acid, right? So if this is a conjugate base, then it should have H4O positive, 2 positive should exist, right? That means it is not a conjugate base of anything, right? So answer is A only, right? So concept is very simple. Concept is general concept that you have seen in your lower classes as well. But the same question exactly you can see in your NEET exam, right? Very simple yet very important question. Okay. What about this one? The bond formed which is non-directional in nature. What is that bond? Ionic bond, covalent bond, dative bond? None of these. Ionic, covalent. Beta, what is dative bond firstly? Dative bond is coordinate bond, okay? This is coordinate bond, right? Coordinate bond is definitely directional. You know there is a direction to this, right? Coordinate bond is definitely has a direction. Ionic bond is non-directional. Why? Because this is nothing but this is a force of attraction. This is nothing but a simple force of attraction, right? A positive charge, a negative charge, they are getting attracted. There is a force of attraction involved, right? So that is why this is non-directional. Covalent bond is also directional because more is the electronegativity. For example, you have A and B. Out of this, B is more electronegative. So electrons will be more here. And there is also one direction to this, right? That is why you form shapes and all, right? So this is also direction. This is not direction. This has a lattice structure, right? This has a lattice structure, right? Next is dative covalent bond is found in. Dative covalent bond. As I told you, dative covalent bond means they are talking about coordinate bond. They are talking about coordinate bond. So coordinate bond is found in which thing? It is found in which thing? Ammonia, that is NH3, or NH2CONH2, or NH4 positive, or nitrogen N2. It is found in which? It is found in which? Beta, it is found in NH4. Okay. This to have simple triple bond, covalent bond. This has simple bonds, double bond and single bond. Ammonia has simple structure, lone pair and covalent bonds. This ammonium ion, that is NH4 positive, right? It's like this. It's like this. Okay. It's like this. It has a structure like this. Okay. Okay. Yes. Understood? So it forms a dative bond here. Right, bachcho? Everybody clear? Any problem in today's lecture? Any problem in today's lecture, bachcho? Any problem? So that was a very short, sweet chemistry, bio. I would say that altogether lecture was very long. But yeah, you have done all the parts here. You have done bio, you have done physics, you have done chemistry. And I think all these questions that we have discussed here, these are actually NEET kind of questions, okay? But these are the easy NEET kind of questions that are from the basics, right? So similarly, we will do more sessions of like that. We will do more question practice. So if you want more sessions like this, more questions, you want to solve more questions, then please write it down in the comment section. How you like this session, and do you want us to create more sessions like this only for you? And also share this with your friends, right? So we want you to share this with your friends. We want a lot of students here whom you can discuss with, and also, you know, there will be a healthy competition then, right? Okay. Yes, bachcho. Now here is this one question, right? One question for you. That is, I want all of you to write it in the comment section. One question for you. You need to tell me the number of orbitals, the number of orbitals in the number of orbitals in the number of electrons in d subshell. A very simple question. Okay, a very simple question. Write it down in the comment section. I will strictly wait for your comments on my. I will see that when I will do my lecture of chemical bonding, right? We are going to do chemical bonding lecture, beta. Till now, we have completed the first lecture where we have done Lewis dot structure. So if you have not seen that lecture, please go and watch that lecture, because in that lecture we have started from the very basic thing, like types of bonds, how ionic bonds are formed, lattice energy, Born-Haber cycle, and we have discussed about Lewis dot structure. We have made around 25 structures that day. I assure you that apart from those structures, you will not see any other structure in your exam, right? So after that, beta, we are going to start with VBT, Vesper theories and MOT and all that. For that, you need to see the first lecture also. So still you have time, you have Saturday, you have Sunday. You can go through that lecture as well. And then I don't want you to create more and more backlogs here, right? And one last thing here, beta, that we have very important, that is NEET PDF for you, where we have physics, chemistry, bio, complete syllabus for you, and for practice you have last 5-year question papers. You have video analysis of 2024 question paper, the video solutions for that, right? Then bacha, for revision, if you are preparing yourself for NEET, if you are preparing yourself as a dropper, you are preparing yourself for 2025 and 2026, it's just 10 months away, okay? So mind maps and revision books are there. Then bacha, we have most important NEET PYQs that are repeatedly asked in your NEET exam. Then we have all India rank and college predictor for 2024 students, and the detailed list of medical colleges. This is for 2024, 25 and 26 students. Reason, because if you don't know the target which you want to achieve, the medical college that you want to achieve, it will be very difficult to make a mindset for that, right? Then you have totally free mock test. So beta, all of this is totally free of cost. The link is in the description. You can go check out the link and download it, right? Okay, bacho. Okay then, bye bye, take care, see you all in the next session. Bye bye, see you all.